So good evening, everyone. A very warm welcome to all of you joining us from different parts of the world. On behalf of Krino Forensics, it is my pleasure to welcome you to this international free webinar titled Decoding the Ridges, Exploring the Biology of Fingerprints. We are truly grateful for your presence today and for your continued interest in advancing forensic science through learning and dialogue. About Krino Forensics. Krino Forensics is a student-led forensic initiative committed to making forensic science accessible, engaging, and practically relevant for students and professionals alike. Through expert webinars, academic collaborations, case discussion, and research oriented sessions, we aim to make forensic science impactful and globally connected. So before we begin, a few quick guidelines for the smooth session. Kindly keep your microphones muted throughout the presentation. Please post your questions in the chat box during the session. A dedicated question and answer session will be conducted towards at the end. E-certificates will be provided to all the active participants who attend the full session and complete the feedback form. Thank you for your cooperation.
Now it is my honor to introduce our distinct speaker for today's webinar, Mr. Nicolas Craven, CLPE Latent Print Examiner, C2 Quality Solutions. Mr. Craven is a certified latent print examiner with extensive professional experience in fingerprint identification and friction ridge examination. His work reflects the highest standards of forensic science, accuracy, and scientific integrity. We are truly honored to have him with us today as he guides us through the anatomy, physiology, and biological foundations of friction ridge skin and its vital role in forensic identification. Without further delay, I respectfully invite Mr. Nicholas Craven to begin today's session. Sir, the platform is yours.
Thanks and good morning for allowing me to present today. Can everybody hear me all right?
Yes, sir, you are audible.
Sounds good. Okay, good. I'm going to go ahead and share my screen because I got a presentation our presentation to go over. Let me just pull it up here. Stand by 1 second. All right. Can you see my presentation well as it comes up here? Stand by. Got it. Okay. So normally what I like to do is while I talk a little bit about myself, I've got a poll here for if you all able to take it for me, if it will come up. And this is just asking people just to give me a little bit about their experience within the world of forensic science, hopefully within the world of fingerprints. You can either use the QR code or there is the website you can punch into your browser and then use that code right there to get access to it and answer this question. I just like to see where people are at as far as experience within the world of forensic science and or fingerprints. But just a little bit about me, like was mentioned, I am a latent print examiner. That's the term we use in the United States for someone that does crime scene work in relation to fingerprints found at crime scenes and things like that. I've taught in the United States. I've also taught in Colombia. I'm currently doing my PhD in a university in Switzerland. So I know that sometimes terms are slightly different from country to country in regards to what we refer to as fingerprints or latent prints or things like that. But generally, when I'm speaking of latent prints, I'm referring to a finger mark or some kind of a trace that's left behind from someone touching something that then gets compared to an individual within a case, someone that the investigators, the police, whoever think might be involved. We're comparing that unknown latent print finger mark trace that's found at the scene to somebody that the police think could be involved or at least want to rule that person out. Okay, I'll just give just a few more minutes here for just kind of see if anyone gets a chance to tell me how many years experience and stuff like that they have. But I've been doing this type of work, fingerprint work, since late 2014. So we're coming on 11 years now. I used to work for a police organization within one of the states in the United States, the state of Idaho. I still do some contract work for them. And then I also do some private cases along with trying to do some teaching and presentations and the like. Like I said, I am working on my PhD looking at features within a fingerprint and can we use those to enhance our comparison when we're talking about identifications and the like. But yeah, it looks like most of you have answered your question. So most of you don't have any experience yet. That's good. I'll try to keep this as basic as possible. This class, this workshop, I've given this workshop a presentation like this at least 2 times before at this organization that I'm a member of. It's called the International Association for Identification. And it's an organization that's primarily made up of people that do crime scene, fingerprint work, shoe print work, stuff like that they have conferences every year. This last year was in Orlando, Florida. The next year is going to be in St. Louis, Missouri, within the United States. And it is an organization that any of you could be a part of. And they also have certifications that one can pursue. And that's where my certification comes in, my certification in latent print examination. And so this last year and the year before, I had given a workshop, a presentation very similar to the one that I'm about to present to you. And this one is more geared towards people that are currently doing crime scene and fingerprint work, but it's still just the basics of the anatomy and physiology that makes up what we refer to as the friction-ridged skin. That's the special skin on the bottom of your hands, the bottom of your feet, that has and we'll talk more about this obviously, and I have some images and things like that, but it's that raised ridges and furrows of the special skin on your hands and feet. And when you have sweat or paint or other kind of residue or things like that on your hands and you touch something, that's when you leave that fingerprint, latent print, things like that behind. And so those are the things that we're looking for when we're at the crime scene or when we've developed the latent print and moving forward. And so if you have any questions, like I said, you can put it in the chat, or if you can, I think I might be able to see if someone, because is there a raise your hand feature on Google Meet? I think I've only ever used Google Meet once or twice, so I'm still kind of new to Google Meet. But if anyone does have like a really important question in that moment, try to let me know and I can stop for a second. We can address that. Otherwise, we can always, if like I said, there should be a dedicated question and answer at the end. We can always go back to slides 2 if you need to. So we'll go ahead and get started because it looks like we got 16 of you don't have any experience yet, but then those that do, it's less than 3 years. So that's good. So hopefully with this presentation, you'll learn something new about the anatomy and physiology of fingerprints, and you can use that going forward. See. Oh, here we go. So this is just the title. We're going to be looking at anatomy physiology of fingerprints. And I got one more. This one should be pretty quick too. You shouldn't have to log back in, and hopefully you stayed. And this is just how many experience you have, or sorry, how many courses you've had with anatomy and physiology, just in general, and anything that related to skin, the integumentary system, which again we'll talk a little bit more about that in the future. A lot of times, latent print examiners, crime scene examiners in the United States don't tend to have a lot of anatomy and physiology experience. So it'll be interesting to see how many of you do as well, because it's just something that I think is you don't have to be an expert in anatomy and physiology to understand the skin and how it affects friction-ridged impressions or fingerprint impressions found at the scene. But it also is a good way to have at least an understanding, a foundation of what's going on biologically and physiologically when we're talking about fingerprints. I'll just wait just another quick moment on this. And again, like I said, I use these quick surveys just to kind of get a quick guide or an understanding of where people are at when it comes to this type of stuff. So it looks like at least if you have at least 1 or 2 courses in relation to anatomy, physiology, and related to fingerprints, or at least what I call friction-ridged skin. All right, I think we'll move forward. This is just kind of, like I said, just to get a gauge of where you guys are at when it comes to this kind of stuff. All right, so these are going to be the topics that we're going to address in some form. It does say exercises at the bottom. I think I removed those, and I failed to remove it from this slide. I usually do when I do this workshop. I do it for about 3 or 4 hours, and then towards the end, there's a few activities that students will do. Since this is a virtual format, it's a little bit more difficult to do those exercises, so we won't do those. And then also, I've removed the common errors that latent print examiners see, but everything else will be in this presentation as far as like the basics of it. We'll talk about the organization levels and tissue types. We'll go into anatomical terms. Again, it'll just be brief overview of each of those areas. We'll talk briefly about organ systems. The heart of it will be talking about the integumentary system or skin, breaking it down from thin skin versus thick skin. Then we'll talk about the two main areas within fingerprint examination or fingerprint comparison that allow us to make the conclusions that we do, and that has to do with, the persistency of friction-ridged skin, and then the selectivity. And again, we'll dive more into that as we go. And then there'll be some part about what happens when you damage your skin and then how it repairs. And like I said, the last two bullet points there we won't cover in this. But if you do have questions about those, you can always bring those up in the Q&A. Okay, so just like with anything within biology, there's different organization levels or different ways to view things. And so at every level, you've got that builds upon the next level. So we start with the chemical level, and even before that, it could even be the atomical level. And so you just have the chemical level, which goes into the macromolecule level, driving in the cell, tissue, organ system, and then all the way into the organism. And so these are all the different levels, the building blocks that then move from one level to the next, right? And so when we're talking anatomy and tissues, there are four main tissue types within the body, and these are throughout the entire body, not just when we're talking about skin. And there's connective tissue, epithelial tissue, muscle tissue, and nervous tissue. Connective tissue is things like bone, and actually blood is a connective tissue. Epithelial tissue, which that's what we'll talk about more today, is a lining tissue that lines the skin. It lines organs and various other things within the body. Muscle tissue is pretty self-explanatory. It's the tissue that makes up the muscle, whether it's cardiac muscle in the heart, skeletal muscle that allows us to move, and then other muscle types, involuntary muscle that allows us to swallow and various other things. Well, I shouldn't say just swallowing. Swallowing is you have some control over swallowing, but once you swallow something far enough down, you have involuntary muscles that take over. And then nervous tissue, again, that's relatively self-explanatory. That's the tissue that makes up the nervous system, the brain, the spinal cord, and the like. But these are the four main tissue types. Like I said, we'll focus more on the epithelial tissue, and there's different versions of the epithelial tissue as well. You have and it's typically based on how many layers that epithelial tissue is, and then also the shape of that tissue. And you can see from the top line here. Hopefully you can see my mouse moving. It's the simple squamous cells, the simple cuboidal, and the simple columnar. They're usually one layer thick of cells, and then just like I said, based on their cell shape is where they get their name. And then from there's stratified levels where you have multiple layers of these cells, whether it's stratified squamous that again make different layers up to the top, stratified cuboidal, and pseudostratified columnar. The pseudostratified columnar are a special one because they're not quite more than one layer, but just how their shape is, it looks like there's more than one layer. You can kind of see that from the image as well. That it looks like there's more than one layer. Everyone. Oh, never mind. Sorry, I'll keep going. And then there's also these transitional cells, and we'll talk a little bit about the transitional cells too in a minute. And how that plays into friction-ridged skin or skin on the fingerprints. And those are just different layers of cells that are going through different changes as they're moving upwards. So there's like a base layer that then creates the next layer up during mitosis, and then they continue to move up as they go. And like I said, we'll see an example of that in friction-ridged skin here in a moment. So then anatomical position, that's going to be in this orientation where when someone's either lying on their back or standing up, your palm is going to be open like you see in the image right here. Because if you were to rotate your arm this way, the reason we have it like this way because there's the two bones in your body right here. You have the radius and the ulna. Those are the two bones in the forearm. When you have your palm open, they are parallel to each other. If you were to rotate your arm over, then now the radius, let's see here, the radius flips over the ulna, and so now they're not parallel with each other. And so we like to have things in anatomical position so that bones are in the way that they always are. And when we know that we're talking about orientation and where one thing is to the other, we want to know that we're talking about anatomical position. It's always going to be in this kind of a format with the palms open head up, chest up, and all that kind of stuff. So like I said before, we'll just briefly go over some of these anatomical terms just to have things understand what we're talking about when we're using different things and orienting ourselves. And these become very important because they're precise in the sense that when I say anterior, you know what I'm talking about versus posterior. And they are universally accepted. So not only will you see these in anatomy physiology, you'll start to see them in the medical world and even in anthropology. So I studied anthropology in college, and those terms were also very important for that as well, and also for this. So we have terms like anterior or ventral. They're both the same thing, and that means towards the front. And you can see the front of the body would be anterior. Posterior would be the opposite of that. Posterior or dorsal is the back side of the body. So you can't quite see it in this image, but it's the back part of your body. So when we're talking about your face, for example, you'd say your face is posterior and the back of your head is posterior. Medial refers to towards the midline. So if you can imagine an imaginary midpoint in your body that runs down through the center of your body, anything that goes towards that midline, whether it's on the right side or the left side, that is medial. And then lateral will be the opposite. Again, you have that midline, and anything outside or moving away from the midpoint. So it's like midpoint out or midpoint out on either side is lateral. Okay, so now that you know that if I say something like, for example, if in relation to my nose, my ears are lateral to my nose, and then vice versa, my nose is medial to my ears. So that's just a way that we can precisely and hopefully understanding wise know what I'm talking about with in regards to orientation or where something is related to something else. And of course, we have a left and a right side that, like I said, that gets then divided by that midline, that medial line that then pushes out. Also within the body, we have two skeletons, so to speak, and there's an axial skeleton and an appendicular skeleton. An axial skeleton consists of your trunk, so it's like your torso up to your head, and then your appendicular skeleton is your arms and your legs. And the reason this becomes important is because there's some directional terms that we use in relation to the axial skeleton versus the appendicular skeleton. So as you can see right here, so superior or cranial refers to towards the head, and then inferior or caudal refers to towards your tail or towards your bottom. When we're talking about the appendicular skeleton, we're talking about proximal, which is towards the middle, and then distal means away from the origin in this situation, or away from things. And so when we're talking appendicular skeleton, when we're talking about your hand or your arm, distal is referring to going away from your body or going away from your shoulder. So if we're talking about your hand right here, so from your wrist, your fingers are distal to your wrist, and then the other way around is your wrist would be proximal to your finger. So proximal means close to or towards, and distal means away. Now you only use proximal and distal for your arms, legs, feet, and things on your appendicular skeleton. You would use superior or inferior for that axial skeleton. So it'd be like your head, your chest, your body, your trunk is where you would use superior and inferior. So you wouldn't say, for example, you wouldn't say my elbow is inferior to my shoulder. You would actually say my elbow is distal to my shoulder. Okay, so that's where that becomes important, is understanding the difference between your axial skeleton versus your appendicular skeleton. Okay, and here's just another representation of that, just with colors. Your head and your trunk, or your body, your core, is your axial skeleton, where you have your you can see right here your arms are going to be distal versus proximal. So distal versus proximal. So your hands would be distal to your shoulder, whereas your head would be inferior to your chest, for example. Okay. A few more directional terms. We also like to use superficial and deep. Superficial means towards the skin surface. So as you're coming to your skin, that's superficial. Deep means within or away from your skin surface. So if you can see from the image on the right here, it's just kind of a cross section cutout of a chest area with some ribs. And so you have a bone. That bone would be deep to the muscle tissue, which would be deep to the skin. And then the skin would be superficial to all that. Does that make sense? So you've got the skin, which would be the most superficial part. And as you work your way in, that is going to be deep. And on the left here, there's an image of some skin that I had taken a few years ago when I was teaching an anatomy class. Is as you go towards the skin surface, it's superficial. As you go away from that skin surface, it's deep. And so that becomes important when we start talking about the different layers of skin, which we'll get to in a minute here. Is one is going to be superficial to the other, and the other is going to be deep to the other, in that kind of sense. And that's just a way that we can orient or display or discuss where something is in relation to that other thing that we're talking about. Okay. And then just in relation to fingerprints or palm print cards, when we're taking these, it gets a little tricky because it's slightly different. Is that when you've got the when you put ink on the hand and then set it down on the paper, remember anatomical position has the palm open and down, so lateral would be towards the thumb and medial would be towards that little finger. But then when you put your hand down to make that impression, it switches it a little bit. So it would be in this orientation. So the thumb is lateral because it would normally be out, and the little finger will be in. But then when it touches down on that surface to make that palm print representation, it gets flipped that way. But the distal and proximal stay the same. Okay, I think there were a couple questions that popped up. Do we want to talk about those now, or are those for later? I'm trying to see, can I see?
Later.
Let me see. Okay, stand by with me one second. Let me just take a look at these questions real quick. So there's a couple questions about identical twins. We'll talk about that in a few slides coming up. So I'll hold off on that one for just a minute. That's a really good question, and yeah, we'll hold off on that one for just now, and we'll come back to that because I have a couple slides coming up here that we'll talk a little bit about that. That's where we'll talk about the selectivity part of fingerprints. So hold that question for now, and hopefully I'll answer it when we get to it. Just some finishing up with some of the more terms here in regards to directional terms. We also have these different planes, and there are three of them. There's the sagittal plane, a coronal plane, and a transverse plane. What the sagittal plane does is there's a suture in your skull that's called the sagittal suture, and so it basically runs in this direction. Hopefully you can see my head. You can see me touch my head here, and what that does is it separates the right side from the left side. So you can see hopefully you can see right here where it says sagittal plane, that it basically cuts the person in right and left halves. Okay, and they can be equal halves or they can be unequal halves. So a mid-sagittal plane would be directly down the middle of the body and separating into two equal halves. A parasagittal, it looks like I have a misspelling right here. There should be the letter A in between here. I apologize for that. That could be done in unequal halves. So if like we were to drop a line like more here as opposed to towards the middle, or like cutting through my shoulder this way, it's still sagittal because it's going from right and left sides, but it would be an unequal one because it would be cutting me through my shoulder as opposed to the middle of my body or the middle of my head. Coronal sutures, coronal plane. There's also a coronal suture, which runs in this orientation on the head, and that's where it's going to divide the body into anterior and posterior parts, or front and back parts. And that one usually is going to be right down the middle because we're usually thinner in this we're thinner in this direction than we are in like this direction. So usually you don't do it anything but just like straight down the middle. But it could be like you could just take a thin slice from the front. Then there's transverse. So these are also called horizontal planes, and so they're just going to be cuts or sections going in this direction horizontally and cutting things up. If you've ever seen any kind of images from an MRI or other kind of imaging type system in medicine. That's what we're talking about when we're talking about these planes, is we want to cut the body imaging wise into small sections so we can look at various areas within the body, within the hands, the feet, the heart, those kind of things, to kind of see if we can notice anything that's out of the ordinary. So we're going to use these planes, whether it's sagittal going down to right and left halves, coronal cutting down from front and back, and then the sagittal is going to just cut up into various sections going from your feet to your head. Okay. And then when it's done, usually transverse planes are done in cross sections. That's what I'm referring to when we're doing imaging with a MRI machine or a CAT scan or things like that. You're breaking up into different slides. Okay. Looks like we've got one more question. Let me just look at it really quick right here, see if there's anything else. Another good question. Let's hold that one to the end as well. We'll address that at the end. Okay, so moving forward. Okay, and here's just some, here are the anatomical regions. So we like to break the body down into specific regions so we know when we're specifically talking about, let's just say the arm, we're talking about this would be this area would be the forearm versus the arm. And so this way we're being more specific. We're being, specifically pointing to, looking at your chin, that would be the mentis. And so you can see from this this image, it has the more anatomical biological term, but then it also has the more common lay term. So like chin is an example of that. And then and you'll see in parentheses, it'll have the adjective that we would use. So you would say something like the axillary region, which is in the armpit, or you have the oral region, which is around the mouth. So so this is the oris or the mouth, but it's the oral region. And the same thing with your hands. So this would be, if you look at the chart right here, we're talking, this is the palma, but it's the palmar region. And then you have the digits or the digital region. So and this would be the palm or the palma. And it'd be the same thing for the feet. If you look down at the feet, you have the pedal region, which is the foot. But then also, if you look, it says there's the plantar region. Hopefully you should be able to see it right here. The plantar region, or the planta, is the bottom or the sole of your foot. So then we know when we're talking about the palmar, we're talking specifically about this area versus the plantar, which would be on the bottom of your foot. Or the digital region would be your fingers themselves, or the digits. Okay. That's about it with that. We'll move on. And then just lastly, these are just all the organ systems within the body. We won't go into anything more than just briefly. This is what they are. There's several of them, from the skeletal system to the muscular system to the cardiovascular system. The one we're going to focus on the most is in the upper left, the integumentary system, which we'll talk about here in a moment. But just they all work they all work together for your body to function appropriately. Because if they didn't, then that's when you'd start having problems. You would need to seek out medical assistance and things like that. But there is some interchange or connection between all these areas. But we're going to primarily focus on the integumentary system. Integumentary system, or the skin, is what we would call it within biology. And so here's a couple cross sections of, you got thick skin on the right, on the one side, on the left, and then thin skin on the right. So skin's primary function is to support, and by supporting it, it protects you. It helps regulate your fluids as well as your temperature. It helps with vitamin D synthesis. So how it protects is it's a barrier that doesn't allow certain things to get in your body, because if we didn't have that barrier for skin, we would have all kinds of, or even more diseases and viruses attacking us than what we probably already have to deal with. And anytime you have a cut or a wound, that then allows germs and various bacteria or viruses and things like that to enter your body. So as long as that skin stays intact, it protects us quite well. Then it also helps us regulate our fluid levels. So what this means is when you're outside and it's really hot, your body starts to sweat, and you develop sweat on your skin that helps cool you down. Or there's also blood vessels within that are just directly underneath your skin or deep to your skin, that those blood vessels can either constrict or expand, that then allows heat to either be retained within your body when your blood vessels constrict, or they can open up and allow heat to dissipate out through your skin. And then vitamin D, we need vitamin D from the sun and various other things to kind of help with synthesis, and it helps with the synthesis so we can develop, helps with calcium buildup and things like that, and help with bone growth and whatnot. There are also, so the integumentary system consists of skin, but it also has a couple accessory, well, more than a couple, a few accessory structures, and that includes hair. There are also glands, which we'll talk about those glands in a minute, and nails. So the skin is considered the largest organ of the body. If you think about it, it has quite a large surface area, so it covers the extent of your entire body, and it consists of all the tissue types, from, of course, the epithelial, which we talked about, and we'll talk a little bit more about that, connective tissue, muscle, and the nervous system. So all those tissue types are within the integumentary system. There's a question. Let me just double check. Well, we'll talk. We can keep that one for a minute here as well. This question right here is an interesting one. I don't know if I would quite answer it. This one's asking, how does sweat chemistry affect the quality and longevity of latent fingerprints? That's a really good question that I don't know if I have a good answer for you. There is some current research out there looking at how to help determine how long a fingerprint has been on a surface, and they are using things like sweat and the breakdown of the residue and amino acids that are detected within that fingerprint. I hope I'm answering your question the way you're asking it, but that's a really good question that I don't know quite how to answer it. Because I would think the sweatier someone is going to be, the longer that fingerprint might retain on the surface, and it might be a better quality fingerprint as well. But also the quality of the fingerprint also has to determine, or also depends on the chemicals we use to have it become more visual, or to visualize it, and how well that fingerprint gets preserved. I hope I answered that question. If you want more of a, if that didn't quite answer that question, let me know. We can discuss it more at the end. But that was one I thought was somewhat relevant to what we're talking about right now. And there's a couple other ones that I think we'll wait for the end as well. Here, when it talks to collecting and preserving latent prints, we can definitely talk about that at the end. So just getting back to this, there are three principal layers of the integumentary system. Some will say there are only two, but I like to refer to them as three, where you have the epidermis, which is the most superficial, the dermis, which is in the middle, and then the hypodermis, which is the deepest part of the integumentary system. The hypodermis is the one that usually gets left out, because it's kind of a transition between the integumentary system into the more deep areas within the body. So it doesn't always get added to the integumentary system, but I think it's worth adding it in there and talking about it as part of the integumentary system, and things that you should have at least a rough understanding of when we're talking about fingerprints. And like I said, there are the two main types. There's the thick skin, which we'll talk about, that there's no hair in thick skin. So if you look at your hands, there's no hair there, likewise on the bottom of your feet, but there would be hair like on your arms, or obviously your face in some capacity, or your head and things like that. And so then that's the thin skin versus the thick skin. We'll talk more about that as we go. So epidermis, like I mentioned, it's the most superficial layer of the integumentary system. It's superficial to the dermis. We discussed this in terms of layers or strata, because it's stratified squamous cells. Those are those flatter cells that we talked about at the beginning that then layer upon each other as we're going from deep to superficial. And the majority of the cells that make up the epidermis are keratinocytes, and they're called that because of the keratin that's in them. And we've recognized about 20 different types of keratin, and this is keratin that is within the skin cells throughout the entire body, not just the skin of fingerprints. But then there are some of these keratin types, excuse me, that are specific to the friction-ridged skin or the fingerprints. So ones like K6, K9, K16 are part of the skin that's on your hands that make up the fingerprints. So the K9 is a keratin that's used in primary ridge, and we'll talk about primary ridges in a minute. And then K16 is part of the secondary ridges, and again, we'll talk about that in a minute. And the K9 is a specific one because it allows the skin to be more durable, whereas the K16 then allows the skin in that area to be more pliable. Okay, primary ridges, again, we'll talk about that in a minute, but the primary ridges are underneath where the ridges on your skin come up, and then in the furrows or the valleys that's made from going from ridge to ridge, the valley in between the two ridges. I'll show you an image of that here in a minute. That makes that area more pliable, and I'll bring that up when I get that image up. And then also the epidermis is avascular, meaning there's no blood supply within the epidermis. The epidermis and the cells within the living cells within the epidermis get their nutrients and their nutrients, right, from the dermis, and it diffuses upwards. It diffuses from the dermis superficial to the epidermis. And the keratinocytes, what we talk about, that's the most abundant cell in the epidermis. I'll talk about the other ones here in a minute. There are 5 layers within the epidermis that make up the epidermis. Excuse me. The most superficial one is referred to as the stratum corneum, or it's also called the horny layer. Like I said, it's the most superficial. This one consists, it's several layers thick, consists of anywhere from 15 to 30 different layers of skin cells, and those skin cells, the keratinocytes, are dead at this point. They've gone through their entire life cycle, and at the surface of your skin, that's why when you say you are scratching your arm or you bump up against something and you see some skin cells come off, that's the dead keratinocytes that are part of that stratum corneum. Okay, they're flat, dry, flaky, and they are waterproof. There's an oil that's part of the skin, as well as when they link together and tightly hold onto each other, that provides that waterproof layer, so that when you jump in the water or take a shower or a bath, whatever, you don't get water to come into your body, because that could then bring diseases, bacteria, all that kind of stuff. And so that provides that waterproof protection that I was referring to before. Directly underneath the stratum corneum is the stratum lucidum. This is typically a single layer. It's translucent. These are cells that are also dead, that have gone through their life cycle. You tend to only find the stratum lucidum in thick skin, or that skin, like I said, that's on the bottom of your hands or the bottom of your feet, that the friction ridge skin that I'm referring to. You would not find that in thin skin. Directly below the stratum lucidum is the stratum granulosum, also referred to as the granular layer. Here is this is the layer where the skin cells of the keratinocytes start to go through their cycle, their dying, I guess, their death cycle, if you will. So more deep is where of the granular layer is where the cells are alive and they're transitioning, and the keratinization process is happening, where the cells start to go through their apoptosis, or their dying cycle, to become that flat linked cells as they're moving towards the top. Okay, the granular layer gets its name because there are granules within the cells right here at this layer. And then, like I said, that's where they start to go through their programmed cellular death. So as you're towards the bottom of that layer, towards the top, they're starting to die off and become the flat, flaky cells that we know of on the surface of your skin. Directly below the stratum granulosum is the stratum spinosum, also called the spiny layer. That's just because of the way the cells look at this stage. They look more spiny or have spines sticking out of them. Cellular metabolism happens at this level. You'll see melanin, or the pigment from our skin, or what colors our skin, is located in this layer, and that melanin comes from melanocytes. We'll talk about melanocytes here in just a moment. And then there was an Italian, I guess he was considered an anatomist from a few hundred years ago. His name was Malpighi. he was the one that I believe identified this layer, and so that's why it's called the Malpighi layer. And I don't remember if he was the first one to use a microscope or not, but he was an anatomist, a biologist from, I want to say from the year 17 something. So a few hundred years ago, when he was doing his research and exploration on human skin and anatomy and things like that. And so he was given the name, or this layer was named after him because of that. And then the last layer, and maybe the most important layer of the epidermis, there could be some debate on whether this is the most important one, but is the stratum basale, or the basal layer. It's the deepest layer. It's also referred to as the stratum germinavatum. It's a single row of cells, but it's all. This is where those, if you remember from that one, the initial slide that I was talking about, the epithelial cells, when I was talking about the transitioning cells, they're also referred to as the transient amplifying cells, and I have an image of that here in a minute. But it's a singular layer, but there's also like a supra basal layer, or just another like small, I guess, half layer, if you want to call it that, that's just above it as these cells are going through mitosis and creating new cells. So all the cells that get created on the epidermis start from the basal layer. There's a set of cells that are linked together, stuck together. We'll talk more about this in a moment, that stay there the whole time, and this allows the skin to stay intact and remain the same throughout your life, provided you don't have any damage or cuts or wounds or things like that. And so all your cells that generate from this layer move up towards the top, go from the stratum basale all the way up to that superficial layer, the stratum corneum. And also at this layer, you'll find melanocytes and Merkel cells. Merkel cells are the nervous cells of the epidermis that provide tactile abilities or touch abilities. This layer also is part of the basement membrane. So there's the basal lamina and the reticular lamina. We'll talk a little bit more about that in a minute. And it also provides the filter or the connection with the dermis. So that's where we get that's where those cells that are still alive, especially the basal layer and the spinal layer, get their nutrients or the things they need from the dermis, as it comes through the basement membrane and through the stratum basale, going upwards until those cells start to die. And then, like I said, mitosis, cell division, happens at this layer. I won't go too heavy into this, but mitosis, if you've taken any biology classes before, it's just mitosis is just the process with which cells create new cells. And so the cell will start going through this process. It will replicate the DNA. The DNA will get separated on two sides. They'll get pulled apart. The cells will start to break apart, and then eventually they'll become two cells. And this is just the cycle that it goes through. When we're talking about the basal layer, the cellular mitosis, at that basal layer, and again, we're talking about just the epidermis here, is you have cells at the very bottom. So you can see at the bottom of this image, the pink cells is where it connects to the dermis. The blue cells are part of the basal layer. And so those cell A's, where it says A, the letter A, all those cells stay there and continue to produce more cells as your body needs it. And so as it's going through cellular mitosis, like I said, the DNA gets replicated, it starts to get pulled apart, and eventually two cells get created. So you have a cell A and a cell B. But B stays part of the basal layer at this point. It's become part of what we call the suprabasal layer, and it's part of those transitioning cells that we were talking about before. And then it still continues to develop as more cells underneath it. So then A creates, again, goes through that mitosis again, replicates the DNA, it gets pulled apart, creates a new cell. In that case, it would be cell C. And as that new cell C moves up, cell B moves up, and then eventually moves up into the stratum spinosum, and it starts to go up that way. Okay, so at the basal layer, those cells stay the same. They are connected to the dermis, and as long as they are intact and continue to produce cells, all the cells going forward from that basal layer to the stratum corneum are going to remain the same. The structure is going to remain the same. Okay, and we'll dive more into that in a moment here as well. And then here's just the image that we were talking about before, the different layers from the basal layer all the way up to the stratum corneum. And from deep to superficial, an average keratinocyte will take about 30 days to reach go from the basement, or sorry, the basal layer all the way up to the stratum corneum, and then slough off or fall off and not be part of the structure anymore. So approximately every 30 days, we have new skin that shows up in the epidermis. So every 30 days, you have all the skin cells minus the basal layer. Remember, those stay the same. Every other skin cell within the epidermis gets renewed every 30 days. Okay. The amount of those keratinocytes is regulated. So we need to make sure, your body needs to make sure that, say you get scratched or you rub off some of the skin on the superficial layer, your body is interconnected. So all these cells are interconnected with each other, and it reaches down to the basal layer and says, hey, we lost a few skin cells on the surface. We need to start making some more so that more gets pushed up as we go. So what that's referred to as homeostasis. Our body is very good at regulating body temperature or pH in the blood and things like that. And so we need to make sure homeostasis is what the body does to keep our body at the normal levels that we're supposed to be at, skin being one of them. And so how the homeostasis works, one way is through what's called the negative feedback loop, So if you look at the diagram on the left, it's talking about a stimulus or something that gets acted upon or that increases to a level that could be dangerous for the body. And so when that stimulus reaches a certain level, there's a sensor that detects it. It gets sent to a control center, which then controls what's called an effector. And that effector then comes around and then changes that stimulus back to the levels that we need it to be at. And so an example of that is body temperature. So our body temperature is typically at 37 degrees Celsius. If it starts to increase above 37 degrees Celsius, the nerve cells in the skin and in the brain detect that. It tells the control center in the brain that our temperature is rising. We need to do something about it. And so then that control center then has an effector. In this case, when we're talking about temperature, it has the sweat glands throughout the body produced sweat that then comes back around, and cools the body back to that 37 degrees Celsius. So if we start ticking up to 38 or 39 degrees Celsius, the body recognizes it and then does something about it to then bring that temperature back down. And so it's the same concept when we're talking about, like I was saying before, with the superficial skin. As it starts to lose, or say you have a really superficial cut that only affects the epidermis, and you lose a few skin layers off the top, your body is smart enough and knows that because of the connections it has from all the layers of the skin, that you just lost some skin off the top of the epidermis. You need to create more cells that then work their way up to then fill in that area. Okay. Here are just a few of the other non-keratinocytes in the skin we've talked about. I think we've talked about all of them. Oh, we talked about all of them except for Langerhans cells. But melanocytes, like I mentioned before, they produce melanin. That's the pigment that gives our skin the color that it has. The amount of melanin and also the type of melanin depends on how dark or light that skin becomes. And then, melanocytes provide or give the melanin to the different keratinocytes released around it, and it helps protect it. And it produces the vitamin D, like we talked about before, that helps with protection of the skin cells and things like that. Merkel cells, part of the nervous system, I mentioned before, they're responsible for touch. They allow us to feel things when we touch with our skin in our hands. And then Langerhans cells are part of the immune or lymphatic system. They help fight off invaders if there's any kind of bacteria or virus or other kind of germs that are invading the epidermis. These Langerhans cells will seek them out and help protect them. They're also dendritic cells, and so you can kind of see how they start to branch out and can reach other areas. And so they will help protect us from bacteria and other bad things that are trying to get into our body. So now we're going to move on to the dermis. The dermis has two main layers. There's the papillary layer and the reticular layer. Okay, so remember, the dermis is just deep to the epidermis. In the papillary layer, it consists of loose connective tissue. It also has these dermal papillae, which you can see towards the top right there. It's the little pegs that are coming down from kind of like those more pink purple colored cells. I think, hopefully you can see my mouse. It's this peg right here. Those are that's the papillary layer. And the pegs help attach the dermis to the epidermis. Or sorry, no, this is still part of the epidermis. My apologies. This is the dermis right here. So this area right here is the papillary layer, and so this just creates more surface area for the epidermis. This is still part of the epidermis right here to connect to the dermis. The capillary layer, or sorry, the capillary loops, sorry, excuse me, are in the papillary layer, and they circle up towards the pegs, or the part that come down from the epidermis, but then that's where the blood supply comes from to then reach the epidermis. The cells within the papillary layer of the dermis are called Meissner corpuscles, or tactile corpuscles. They're also helping us with fine touch and feeling and whatnot. I mentioned before it's the dermal connection between the basement membrane, or that area around the junction between epidermis and dermis. And I also mentioned this as well, it helps mold the connection between the epidermis and the dermis by creating more surface area. So when you have these pegs that go up and down, it's creating more surface area to then allow that stronger connection between the epidermis and the dermis. In the reticular layer, that's the part a little bit further down, so it's a little bit more deep from the papillary layer. It consists of dense, irregular connective tissue, and that's where it provides strength and resilience. So that when your skin gets pulled in multiple different directions, your skin retains and holds on to itself. There is another connective tissue that's somewhat similar to this. It's called dense regular connective tissue. And you can kind of see from this image, you see kind of how it all runs, even though it's got some wavy appearance to it, it basically runs from one side to the other in like a linear direction. Whereas in the reticular layer, you can kind of see down here in this image just below the one I just showed, is there's no real linearity to it. It goes in multiple directions, and so that allows it to be pulled in multiple directions. Whereas this one I'm showing you right here with the dense regular connective tissue, it's strongest when you pull it in its linear form, but if you were to pull it perpendicular to that line, it would not be able to withstand. So that's why the reticular layer in the dermis needs to be that dense irregular tissue, because your skin gets pulled in multiple different directions between, going about our daily business. You would see these dense regular connective tissues in things like tendons and joints, say for example, like in your knee, like your knee when you're walking, that your knee can be pulled in one direction, but if you were to hit your knee from the side, you might tear some of your tendons, like your ACL, your PCL, or things like that within your knee, which is not good. And anyone that plays any kind of sports, whether it's soccer or cricket or rugby or various other those sports, if you hit your knee from the side, like if this were to be my knee, if you were to hit it from the side, you would have a very bad day and likely need to have some kind of surgery to correct those type of injuries. Whereas with the reticular layer, where it has that dense connective tissue, it can be pulled in multiple different directions and not affect it. Okay. The reticular layer also connects with the hypodermis, and you will find in this area the sweat glands, the sebaceous glands, more blood vessels. You'll find laminated corpuscles. That's another pressure sensor. And then there'll be nerve endings that helps with sensing pain. Moving on to the hypodermis. So this is the deepest layer. It's also called the subcutaneous layer. It's primarily composed of adipose tissue. And here it helps with energy reserve. It cushions the skin. It helps with mobility over other structures. It contours the body. It's an insulator. Kind of just helps protect you overall and gives shape to your finger. So you can kind of feel a little bit of some fattiness. And that's going to be from that hypodermis that gives the fingerprint some shape. So when you're touching something with your finger, if you didn't have the fatty hypodermis layer, it would just be like a flat skin with bone underneath it. So that just kind of helps get it some shape, that roundness that we know when we look at our finger. Let's see here. So yeah, moving forward, it connects with the dermis by some interlocking fibers that just kind of interlock with each other, so they hold tight, and it will also share some blood vessels with the dermis. Okay. Briefly talking about these accessory structures, also called appendage structures. So within the, like I mentioned before, within the reticular layer of the dermis, you'll have different glands, and there's two main types of exocrine glands, and then there's also the apocrine glands. And the apocrine glands are not pictured in this image, but the exocrine glands are. There's two types of those. There's sebaceous, which produces sebum, which is a more oily structure that then it's the oil that protects the hair and coats the hair. You do not have any sebaceous glands within the friction-ridged skin or the skin on your hands and feet. You only have the sweat or eccrine glands, and those eccrine glands produce the sweat. And like I said, that's the only accessory found in thick skin is the eccrine gland. And there's many more of those sweat glands on your hands and on your feet than there are throughout your body. As far as per square millimeter, you'll find more sweat glands per square millimeter on your hand than you would on other part of your body. So you'd still have the sweat glands on your arm, but they're much more dense in your hands. Apocrine glands, you tend to find them in the pubic area as well as the axillary layer, and they also produce an oily, fatty secretion. Hairs, we won't go into too much in this. They have a shaft and a root. Dead keratinocytes reach the surface as part of the shaft, and then the root is embedded in the dermis. Without a root, you need a DNA tag from the root if a hair was pulled to be able to do a DNA analysis on that. But just finding the hair in the shaft doesn't always help with DNA structures. And then nails, again, I won't go too much into that. They are also dead keratinocytes. So they're made of, it's a different type of keratin, but it is keratin on your nail. That's the same, not the same keratin, but a similar keratin you would find in your skin cells. So I have this video that hopefully it shows well. And if not, I can send it out to you. Can you all see this all right? Can everyone see the video? And this is just a time-lapse video showing skin and the sweat coming out from the pores. And so you can see right here, all the different beads of sweat right there are from different pores within the friction-ridged skin, which all have their own sweat or eccrine gland. And as you can see how tightly packed or how dense each of those sweat pores are. And then you'll see a little bit more here as it goes. And so this is a time lapse. This doesn't happen this quickly when the sweat beads up. It's just a way to show how that looks. And then this person will touch a piece of glass, and you'll see that at first you can't see the impression that's left behind until you adjust some lighting. See how at first you couldn't see, but then once they adjust the lighting, you can see how the sweat creates that image. And if you can't see the video, I apologize. I'll try to see if we can't get that to you in another fashion. Make sure. All right. And then so here's the sebaceous gland. So the sebaceous gland is over here. It's this the yellow area. It's this yellow area right here that creates the sebaceous gland. Or that is a sebaceous gland. You can see it attaches to the hair follicle. And then sweat glands are down here. So the sweat gland is this structure right here that has all these tubes that then run all the way up into the pore that then reaches the surface of the skin, whether it's for thin skin or thick skin. Okay. And let me know in the chat towards the end here if you didn't get that that video that I just showed, the sweat pore video, and I can send it along to you all, and we can get you that video. And then there's these muscles in the skin, which I think is one of the cooler muscles. I just kind of find it funny because I like the name of it, but it's called the erector pili muscle. And what this muscle does is that when you're cold or when you get frightened or when you're nervous, that muscle then causes the hair on your skin to stand up. So when you get cold or chilled or frightened, that muscle contracts and it causes your hair to stand up on your skin, whether it's like on your arm or maybe like on the back of your neck. So if you ever have that feeling, it's because of these erector pili muscles. I just point that out because I think it's kind of it's a fun name and it's an interesting muscle that its only purpose is to cause the hair on your body to stand up. Like I said, whether it's on the back of your neck or on your arm. So if you ever had that feeling, it's from these muscles. All right, so we're going to talk a little bit about the difference between thin and thick skin. So like we've talked about numerous times, thin skin covers the majority of your body, except for the palmar region or the plantar region, which that's on your foot. Not sure what. Oh, the thin skin in the stratum corneum isn't as thick as it is on thick skin, and hopefully some of these images you'll kind of get to see what I'm referring to with that. But as the top right here, you can see where it says stratum corneum, and it's thick here, but it's even thicker in thick skin. And in thin skin, you have the hair and the sebaceous glands. You're not going to have those in thick skin. And then, as I mentioned before, typically you're not going to have the stratum lucidum in thin skin. It's only going to be the stratum lucidum in thick skin. And moving on to thick skin, like we mentioned, this covers the palmar and plantar regions of the friction ridge skin. I abbreviate that as FRS, or friction ridge skin. That's the special skin on your palms and feet. It has a thick stratum corneum. So I don't know, it's kind of hard to tell, but the stratum corneum is decently thick in thin skin, but it's even thicker in thick skin. There's multiple layers of that stratum corneum that helps create that structure, the mechanical, that helps with it, because you remember you have excessive mechanical stress or stressors on your skin, but it's even more so in your hands when you're grabbing stuff and touching stuff all the time. Because if you think about it, if the skin in your fingers and your palms was as thin as on your arm, you'd have cuts and bruises and wound openings all the time. That's why it's much more important to have that really strong, thick, multi-layered skin cells on your hands and feet, because we're using those all the time. And it tends to be 25 to 30 times thicker in thin skin. The stratum corneum tends to be 25 to 30 times thicker than in thin skin, okay? And then, as I mentioned before, there's only sweat glands in thick skin or on that friction ridge skin, and it's highly concentrated to where you have between 2,500 and 3,000 sweat pores or glands per 2.5 centimeters squared. So much more densely populated sweat pores and sweat glands. And then the stratum lucidum is part of thick skin, and you can see it where I was talking about before, where it's like a transparent layer. So it's a thin layer of essentially see-through cells, or cells that you can see through. So kind of moving a little bit more into how this affects fingerprints on some level is, remember I was talking about ridges and furrows. So if you look, I don't think I have a better image than this at the moment, but if you look on this image right here, you have a pore. Can everyone see my mouse all right? My cursor, I should say. But if you look on this image, the image on the left here, you'll see an opening that's a sweat pore, and then so then the pore opens up into an opening that goes down to a sweat gland. So this structure right here is the ridge, just like this is a ridge. So that's the routine. The canine is primarily found on the primary ridge structure, and that's the one that's down here, that's directly below the ridge. That gives it that durability and strength. Whereas in the furrow, you have that K16. So this is the furrow in between the ridges, and this is the secondary ridge. The K16 allows this area to have more pliability, so it can move and rotate more. So as you grab something and your skin starts to fold, the ridges stay intact, but then the furrows close or open depending on how your skin is grabbing things, and that allows your skin to be more pliable and move around and touch and grab stuff. Or when you're grabbing, like say, for for example, this pen right here, I'm grabbing it and my skin kind of rolls around the pen so that I have a better grip of the pen. And so then the furrow would either close or open more depending on how I'm grabbing it. And so like we're talking about, the primary ridges go all the way down into the dermis, and they connect with sweat ducts and sweat glands down through there. And then the secondary ridges, not quite as deep as the primary ridges, but they do connect with the dermis, and they are beneath the furrows. So primary ridges go down the furthest into the dermis, and then they're below the ridges on the surface, and then secondary ridge formation is underneath the furrows. That's a little tricky that we're talking about ridges and ridges all the time, but just remember, when we're talking about primary ridges and secondary ridges, they refer to the connection between the epidermis and the dermis, directly below the structures on the surface of the epidermis. So I apologize if there's a lot of what seems to be redundant ridge structure. So this is a primary ridge like we talked about, and then this is a secondary ridge. So you can see that there's the primary ridge right here. If you look to the one a little bit more to the left of that one, you can see the duct that goes down into the gland. And then you can see that just really quickly right here, the primary ridge goes down a little bit further underneath the ridge, but then in the furrow, the secondary ridge doesn't go quite down as far into the dermis. Now, also within thick skin, we have these special structures that are called minutiae. There's also pores, but those you can see on if you were to look at your fingers, you likely be able to see some of these minutiae in ridges and furrows and things like that, like we've been talking about. So the ridges is that raised skin that we talked about that creates that line. The furrows are in between, and then minutiae is where a ridge will either start or stop. As you can see right here, there's it looks like a ridge ending where the ridge goes and then stops, versus a bifurcation, which sometimes it's hard to tell the difference between a ridge ending and a bifurcation, where the ridge, as opposed to stopping, maybe this time it opens up into two. And then we also have pores. So you can see the pores showing up right here in the skin. Okay. Now what that looks like on there it goes. It wasn't advancing as quickly. So as you would show up on a fingerprint, so this is a fingerprint. I think this fingerprint is mine, if I remember correctly. You'll see that the ridges show up, especially when you use a black ink, which is typically what we do when we want to create that, what we would call a known standard. So you know who I am. So we're going to take my fingerprints in a controlled manner to create a fingerprint that we can then use to compare to an unknown sample at a crime scene. And so when we use black ink to create that known sample, the ridges are then going to be black, as you see right here, the black line. Whereas, okay, I'm sorry, my slideshow's a little slow at the moment. Here we go. The furrows are that white space or that white area in between the two black lines. And then the minutiae, again, we're going to have ridge endings where that ridge comes and then stops is a ridge ending. So it's the black inked line comes and stops. That's a ridge ending, versus a bifurcation where two ridges come into one, or maybe one ridge opens up into two. Excuse me, that's called a bifurcation. You will also find at times what are called incipient ridges. They can also be referred to as nascent or interstitial rudimentary ridges. These are formations or incipient ridge formations that are not mature ridges. And so in between in the furrows, so in between the ridges and in the furrows, you will sometimes have these incipient ridges that are immature ridges that didn't form as fully as the ridges. And so you will see these sometimes and sometimes not. And they can be person dependent, meaning, I might have incipient ridges and, my friend might not have incipient ridges just through development. The thing to remember with incipient ridges, they can be useful when we're doing a comparison between an unknown sample at a scene versus a known individual in the case. But incipient ridges don't always reproduce, in the sense that if I have incipient ridges on my finger and I touch a surface, because of their immature weaker nature, they might not show up in either the unknown impression that I leave behind or even in my known sample that I took when I rolled my finger with ink. So we had to use incipient ridges and sometimes pores for that matter cautiously during comparison because they don't always show up every time we touch a surface. And that can be, like I said, even with a fingerprint or a latent print found that is seen, or even when we're taking someone's known impression in an ink format. So they can be helpful, but we have to use them with caution during an actual comparison. And if you have more questions about that, we can definitely talk about that at the end. And again, oops, sorry, there we go. My computer sometimes is a little slow with these lines, but you'll see here's, you can kind of see a pore there. They're hard to see. Pores, much like with the incipient ridges, they don't always show up even in an inked impression where I, rolled my finger nice and smoothly and tried to create a really good impression. So you don't always see these, again, on either the unknown impression at the scene or when we do it in a controlled manner. And that pore is really hard to see, and you can tell throughout the rest of this impression that sometimes you see pores and sometimes you do not. Just quickly, these are just the different minutia types that have, this is from a paper that was recently published. You can see at the bottom here, it was published just last year in 2024, that during a survey with a bunch of examiners like me, people were asked what they call these various different minutia type to try to get a consensus or an overall use or naming of these particular different minutia, whether they're simple minutia like a ridge ending or a bifurcation, to a more complex one like an enclosure or a bridge or things like that, to hopefully get everybody on the same page. So when we talk about a short ridge, that's what we mean. So you can see in the box right there where it's talking about the short ridge, is just a smaller ridge you can see right here in there, versus a ridge break. So when I say a ridge break, you know that I'm referring to something that looks like that on a fingerprint, okay? Then there are creases, and you tend to see these not only in the finger, but also across the palm. There are regular creases, again, also in the finger and the palm. The regular creases in the palm are going to be these ones that you can see when you close your hand much easier, that these ones are created before even the ridges were developed or produced when you were in utero or when you were growing in your mother's womb. And then there's also irregular creases, which were either developed early or it just happened to happen due to wrinkling. And wrinkling is sometimes hard to tell between irregular ridges. And so when you look at your palm, wrinkling does tend to happen more as you get older. But because we use our hands and fingers so much and we're constantly moving them and they're getting pressed and pushed and they're pliable, those creases tend to create or become stronger as you get older. And it's hard to tell between whether it's a wrinkle versus an irregular crease, okay? I won't go too much more than that, but again, if you have more questions, we can talk about that at the end. Here's just what creases look like. So this is a regular crease. So this is that topmost crease in your palm. And there's three of them that you can see. You can see these are the regular creases. You have this top one right here, this one towards the middle, and this one towards the bottom. They each have names. I won't go over them at this point. But then you have your regular creases, which you see right here. This one is not a regular crease, one of those more common ones. Most everybody has these same three creases right here. But there is some variation within human population. The irregular creases do tend to be more individual or more selective to that person. I think this is my wife's palm. So that's a crease that's likely specific to her. And then you have wrinkles that, oops, sorry. We could have wrinkles throughout this palm that could be, these smaller ones right here are probably wrinkles of some point or of some sort, okay? I have one more poll question, and let's see if this one works, because this one I think sometimes doesn't work for me. Okay, can we all see this? And so hopefully this question is this thick or thin skin? Just so we're looking at this image, based on what we've talked about before, looking at it and being like, okay, does it have a thick stratum corneum? Are there sweat glands? Are there hair follicles? Are there other things in there? Is this thick or thin skin? Are we all able to see this, and are we all able to get into that survey format thing again, the menti.com? If we can't do this, we can talk about this in a quick second, but you give me a thumbs up or let me know if y'all can see this. Oh, maybe I need to, do I need to start? Maybe I need to start it because it says people are ready. Yeah, this one I had a hard time with the last time I presented it too. So if you want to, if a few of you just want to maybe put it in the chat what you think it is, or if you just get a chance, maybe just throw it in the chat and we'll see what some of the responses are, because this doesn't seem to want to be working for me. Oh, well, oh, here we go. Is that working now? Oh yeah, so what type of skin is this? Is it thick or thin skin? For those of you that are in here. Okay. So most of you said thick skin. I might have to, it's interesting because last time I gave this presentation where this worked, I think there was some confusion too. So I might have to update this image, but this is actually in thin skin, because this right here, I don't know if you I cropped out the line, but this is actually a hair follicle or a gland. I can't remember which. And then the stratum corneum, which is kind of hard to see, is not very big up here. So it is actually thin skin. So I think I'll have to update that from, I think this image is kind of confusing. So I apologize for that. But this is actually an example of thin skin. All right, so now we're going to transition to some of the two main pillars, or the two main reasons why we can use friction-ridged skin or fingerprints and palm prints to identify people or exclude people from, whether it's a crime scene sample, or you want to identify them for like a criminal background of some sort. So the first one is that friction-ridged skin, the skin in your palms and feet, are persistent. And what I mean by that is it remains the same throughout your life. So your friction-ridged skin remains persistent throughout your life with only minor changes. If you remember, we talked about the incipient ridges before. Those sometimes mature and they appear like a minutia. So if we go back a couple slides here, see if it'll let me go back effectively. Come on. If we go back up here, sorry, my computer's a little slow. Okay, so we go back up here and look at these incipient ridges right here. At some point, these might mature a little bit more, become a little bit more robust, a little bit thicker. And even though they are technically incipient ridges, even when they become more robust, they might appear to become a full ridge. And especially like if you look at like this little thing right here, this is probably an incipient ridge. But as, like I said, I think this is my finger, if I remember correctly. As I get older, this incipient looking ridge right here might become more robust and connect here and make this look like an enclosure or something that maybe more so than it is. And so that's a minor change. It doesn't, my skin doesn't change per se, but it might have like a minor change like that. But skin does stay persistent throughout life. And like we mentioned before, likely you'll have increased wrinkles. So this does tend to be more prominent in women. So women do tend to have more wrinkles than men, but that doesn't mean when you see a really heavily wrinkled or a heavily creased fingerprint, it has to be a woman, but it does on the whole tend to happen more in women. And then of course, as your fingers are damaged or the skin on your finger is damaged, whether you get a cut or a wart or other kind of things, that can affect that persistence, right? So this, let me just see here. Yeah, so that's, I'm just trying to think if there's anything else I want to mention on persistence, but that's just basically what it is, that from the time you're born to the time that you die, provided you don't have any damage or disease that happens to your skin, it's going to stay the same. Now, the size, obviously, from when the time you were born to the time you die. So when you're first born, obviously your fingers are a lot smaller, but that friction ridge skin and the orientation of those minutiae and the ridges and the pattern types, that remains the same even from the time you're born to the time you die, provided there isn't some kind of damage or things that affects the structure. Okay. Some of the reasons why this happens, or what keeps your skin persistent, is there's three things, as you can see right here. The cells interlock and adhere to each other and stay together as they're moving up the different levels of the epidermis. The basal layer attaches to that basement membrane. So remember before, that basal layer, the very bottom layer of the epidermis attaches to the basement membrane, and then that basement membrane attaches to the dermis. And so that, again, that interlocks the two layers of the epidermis and the dermis together, and so they don't move. So unless there's any kind of like cut or wound, that will stay the same. And here's a little bit of discussion on that. So the basal layer, the stratum basale, excuse me, articulates with that dermis, or sorry, with the dermis via that basement membrane. And in that basement membrane zone, there's different things that's going on there that interlock with each other to then keep things connected. Okay. So the basement membrane zone has both, like I said, the epidermis and the dermis. It helps filter nutrients from the dermis into the epidermis. And so the area below the basal layer is called the lamina lucida. And then the area that's just above the dermis is called the lamina densa. And so that's part of that basement membrane. They interlock with each other. As you can see right here, this is the basement membrane zone. There's the two the two laminas, the lamina lucida and the lamina densa, that interlock, and there's fibers interconnecting with each other. There's also hemidemosomes. That these are special attachments that project downward into the lamina densa. I have a couple more images of those here in a minute, but they're just like these structures that attach to the cells of the basal layer and then have like tentacle-like things that reach down into the lamina densa that interweave or connect with each other to then create a strong anchoring piece. Then there are also other fibers and anchoring plaques that then, again, help interweave and connect all those fibers within the lamina densa. And so, as I keep mentioning, all that interweaving or all that connection just creates a really strong surface or a really strong connection that doesn't allow the epidermis or dermis to come apart. Because if you think about it, if the epidermis came off every time we touch something, then that would not be good for us. That would probably hurt as well. And here's just another image of that. So up at the top right here, you can see these are, the representation of the basal layers, the basal layer of the epidermis, as it's called right here, the basal keratinocyte. And then you have these hemidesmosomes that connect into that cell. They reach down, these anchoring filaments that then reach up into other filaments or collagen fibers that then interconnect and then allow that interweaving or that connection between the different layers. And you can see right here, there's the lamina lucida and the lamina densa. That's part of the basement membrane. So as long as this area stays intact, your skin, the epidermis, will remain the same going forward for the rest of your life. And we'll talk about damage in a minute and how your skin reacts to that. Now, between the cells, there's also intercellular connections, and these are done by desmosomes and tight junctions. And they're throughout the entire epidermis. This isn't just the special skin, the friction-ridged skin on your hands. This is throughout your entire body. So all those skin cells, no matter where it is throughout your body, are attaching to each other as it's moving from the basement layer up into the dermis. The size of that desmosome does increase as it reaches the surface or as it moves superficially. And those desmosomes are much larger in friction-ridged skin, again, because we want to make sure that those cells, one keratinocyte to the next, they stay connected together as they're moving up to then create that strong bond and maintain your skin structure. Once the keratinocytes reach the surface, those desmosomes do start to break down, and then that's when the keratinocyte or those dead cells that are on the surface of your skin will then kind of slough off or fall off as they go. And then gap junctions are very important structures because not only do they help connect one cell to the next, that allows the intercellular communication. If you remember before when we were talking about homeostasis, is that because those gap junctions, skin cells on the surface, if they start to fall off, other cells just beneath it can notify the bottom and say, hey, we need more skin cells coming up because we just lost some. So that's due to those gap junctions that allow for that cellular communication. As I was mentioning before, as long as the stratum basale and the basement membrane remain intact, the overlaying ridge structure remains the same. When there's any kind of disruptions to just the epidermal layer, so say you lose a little bit off the top, let's say it's a little bit of the stratum corneum, you might just have a temporary line in your finger, but it would just be temporary because it wasn't deep enough. But as it gets deeper and hits that basal layer, if you see on the image right here, if it reaches down into where the yellow tan area is, if it reaches down to there, that's then going to affect and cause scarring to that finger. The shape of the epidermis is the shape under the epidermis isn't as important. It's programmed cellular division, and so how the structure, as far as like the ridges and the furrows, remain the same. Just depends on how frequent the underlying basement or the underlying basal layer, the skin cells underlying the ridge, is programmed for those skin cells to produce faster than under furrows. And so as long as that remains the same and it's in the homeostasis, more skin cells will come up on the ridges than under the furrows, and that maintains its structure of the ridges versus the furrows. So you get that like up and down valley or peak and valley that we see in friction ridge skin. So that it's not so much the underlying structure in the dermis, but it's how often the basal layer reproduces cells underneath the ridges and the furrows. Okay. If you have a question on that, please make sure to put it in the chat, and I'll make sure I address it at the end. So we used to say in fingerprints, and when we're comparing stuff, that the reason we can compare one person to the next is because of uniqueness. There's been some pushback and some change in thought when it comes to this. And I like to use the term selectivity more so than uniqueness. You could also use terms like specificity, variability, discriminability. And what that means is that friction ridge skin, or the minutia, it's much more certain features are much more uncommon than others, and due to that, having those uncommon features in certain orientations, it's much more selective to one person more so than the other. But you can never say that it's absolutely unique to that person, since we can't ever truly determine that friction ridge skin or fingerprints are unique, because we don't have every person that lived in the past, every person that's living now, and every person that will live in the future. We don't know those fingerprint minutia and orientations and ridge flows and ridge structures. We can't say that uniqueness is absolutely a proven thing. Now, we do know, with more than 100 years of studying fingerprints and that special skin, we know that certain ridges and minutia and structures within the fingerprints are highly variable, that we can start diminishing or honing in more on one person over another, and using certain ideas like likelihood ratios, or understanding of that the correspondence between the unknown to the known is much more likely when we assume that it's from that person than from somebody else. Okay, I won't go too heavy into that because that's a whole nother class that gets really tricky to understand. But uniqueness, and I have an excerpt from a very good book that I have the references at the end of this, and I can definitely share the reference as well. It's from a book by Professor Champeau, who's actually my PhD advisor. And basically, he's just saying that we had this misunderstanding of what we mean by unique. Unique, everything is unique, whether it's I touch the same surface 2 or 3 times, each one of those touches is going to be unique, but we can then compare each of those touches back to me. And because of the selectivity, because of the variability that you will witness within my finger, we can then say with a high degree, well, I shouldn't say high degree, within a high likelihood of those fingerprints that I just touched multiple in a row, that they belong to me as opposed to somebody else. but each one of them is unique in and of itself. So using terms like unique is a misunderstood term, okay? And even as far back as the 1960s, researchers were talking about how the structures of the individual ridges are extremely variable, meaning that, like I said, that it's much more selective to one person, but it can't rule out, you know, unique situations or very uncommon situations where the structures look very similar. And I'll hopefully explain a little bit more as we go to here. And then just quickly, I'll talk about this article. This was an article from a couple years ago from some researchers out of, was it Columbia University in New York, that they created an algorithm that looked at fingerprints to determine how unique they might be. And according to them, they claimed that fingerprints are not unique, but they didn't really explain anything or determine anything that we didn't already know. That's why we don't like to use the term unique, because fingerprints are highly variable. And here's the actual article itself, or at least the front page of it. And what they found out is that the ridge structure, the ridge flow for intra-person, or for example, my fingers from finger to finger on my hand tend to be very similar to me. And therefore, because of the similarity between, let's say, my index finger versus my middle finger, that it's not unique. But we kind of already knew that on one person's hand or someone's hand, the ridge flow, the ridge pattern is very similar. And so while I don't think we should say things are unique, there's still the minutia itself. And they didn't really look, and they even mentioned at one point in their article that the minutia are highly variable. But that's the point that we're trying to make, that, or that I'm trying to make at least, and hopefully I'm doing a good job. You can let me know if I am, that I don't care about if it's unique or not. I care about, can I distinguish somebody, a fingerprint from one person to the next? Okay. Again, let me know in the chat later. We can discuss a little bit more further if that makes sense. I don't want to go too heavy on this because, like I said, this is more focused on biology and things like that. But here's kind of an example I want to try to use when we're talking about uniqueness. This is a palm print. I'm pretty sure this is my finger, or my palm. Let's just say on this particular. Like for each impression, a full palm, and then you've got some fingers. Oh, sorry, my computer's slow at the moment. Each one of these touches is unique. It's a full palm, right? And so each one of these two images that just popped up, they're unique. That if I were to do the same touch again with these same two fingers, each of those touches would be unique. Now the thing is, even if friction ridge skin is unique, being able to compare the image on the left back to me is not very easy because of just the quality and how clear, or not clear in this case, the image on the left is. Whereas the image on the right, there's a lot of information. You can see a lot of minutia. You can tell the pattern type pretty clearly. And that one is much more highly variable in the sense that we can probably select it being from me with a much higher likelihood than it being from somebody else. Whereas the one on the left, we would probably never be able to feel good about the fact that it did or didn't come from me if we were going to go that far. Does that, I hope that makes sense, because just the quality of that image is so bad. Even though, again, if we want to say that it's unique, this is my left little finger is the image on the left. That area is unique to me, but this particular impression, we would not be able to link it to me because just how poor the quality of that latent print is, that fingerprint is, as compared to the one on the right. But again, let me know in the comments if that didn't make sense, and we'll go over that hopefully a little bit more. With select, continue with the talk about selectivity, and hopefully this gets into the question that people had before about twins. I'll talk about that here quickly. Is you have what's called genotype and phenotype. Excuse me, genotype is your genetics, what's the DNA, and then when you add that genetics to an environment, it creates a phenotype, or what that person shows. for example, we can use eye color, for example, to where my DNA is coded for me to have, let's say, green eyes, but then something in the environment, maybe some kind of development thing, maybe I didn't eat enough carrots, or I didn't eat enough of whatever, or the sun hit my eyes in the wrong way. That environment caused my green eyes that I'm supposed to have due to my genetics be blue. And so that's what your phenotype is. It's what you show as. And so when it comes to twins and triplets or quadruplets, or however many are from the same egg from the mother, they have the same DNA, their environment is the same, and their phenotype tends to be similar, but not with everything. You know, hopefully you have met twins before that they look very similar, but maybe they have slightly different hair color, or one's slightly taller than the other. Whereas as we're talking about this, it's like, well, if they have the same genotype and their environment is probably the same, their phenotype should be the same, but yet it's different. And the reason for that is, I'll just jump ahead really quick right here, is developmental noise. So there's also randomness from unforeseen events that's happening while you're developing, when you're in the environment, that affects how things are produced, or how it's developed within your body. And that's where fingerprints come in, that even though your DNA is the same, you're in the same environment within your mother's womb. Oh, I'm talking twins here. Fingerprints are going to be different. They can be very similar, like pattern types tend to be very similar, ridge flows tend to be very similar, but those minutia, just because of how minutia are set up or formed while the fingers and palms are producing or developing in utero, the minutia placement is different. And let me just make sure there wasn't anything on this slide that I want to go over really quick. So yeah, it's just localized stressors that during longitudinal growth that affects ridge formation. So as your fingers are growing in utero, and even within the light, well, more so in utero, because your friction ridge on your hands and feet stop developing at like I think it's like the 16th or 17th week of gestational period. So early on, you develop friction ridge skin, and it stops developing pretty early in the developmental stage, and we'll talk about that quickly here in a few moments. So it's localized stresses that cause some changes. And there's an article from 2002, it's referenced right here, that it's Wertheim and Maceo, and that will be in the references at the end of my presentation. I can send that reference to you as well if you need it. But they like to use the example of building blueprints, or when you're building houses. You might have a blueprint for a house that's the same, and you put one house here and the next house here. You're using the same blueprint, but for whatever reason, the materials are different, or when you're putting in the door, a nail goes in a different spot versus in the next house. And so these subtle changes while it's being constructed cause, even though they're very similar in makeup, one house to be distinguishable or variable to the next. And so you can easily determine, hey, this house is different from this house. Even though the blueprints were the same, the idea was to build the same house, but just do the materials present, or where certain nails were put, or just different variations in materials just caused to be slightly different. Okay, and there are immeasurable factors that can contribute to how skin and how minutia and how the ridges form. And again, in that same article, they use another analogy talking about a dump truck. So say you have a dump truck or some kind of a truck that has a bunch of like sticks and various different things in it. If you had two trucks that had, or even if it was the same truck, say you put a bunch of like wood pieces or sticks or boards in that truck, and then you dump it out, they're going to spread around a certain way, one way. And then say you collected all those sticks in again and put them back in the truck and then dumped it out again, it would be different even though you were doing the exact same thing every time. So there's innumerable or measurable factors that can contribute to how things are developed within skin. And then just a couple things that we've learned along the way that could account for how ridge structure and minutia are different from person to person. One way is looking at a Turing reaction diffusion, and I'll talk a little bit about that in a second. And then Merkel cell modeling. And so Merkel cells are those cells in the epidermis that help with touch. And there's some research that shows that as skin is about to start developing ridges, the Merkel cells start to line up along lines of stress, that's where the ridges form, and then minutiae start forming in relation to those Merkel cell modeling, or that Merkel cell formation, right? And just quickly, what a Turing reaction diffusion is, and they were developed by Alan Turing. He was an English mathematician during the 1940s. He helped create what's called the Enigma machine, which the British used to break a code that the Germans were using during World War II. And there's a movie based on him. It's called The Imitation Game. If you haven't seen it, it's a really good one. But he developed a mathematical model that helped explain pattern formation. I have a video for it, but I can try to send a link to the video and at a later date. But basically what happens is you have an activator that creates more activators, and it spreads out and diffuses across an area, but then it also creates inhibitors. And so as the activator is creating and making more cells and creating more inhibitors and more activators of itself, inhibitors will eventually come back around and then cause the activator to stop. And it does that in a mathematical form. And there's a formula that you can use that I don't quite understand the formula myself. But if you're curious, I would suggest looking up the Turing reaction diffusion. But basically, it's just a simplistic, I shouldn't say simplistic, it's a way to determine or show mathematically how patterns are formed, that through these activators that eventually get inhibited by an inhibitor, then spread out and diffuse and create patterns that are that you see across nature. And that's, from zebra stripes to stripes on cows to our fingerprints can all be potentially explained by this mathematical formula that Alan Turing developed in the 40s or 50s, I believe, 1940s or 50s. I won't hit on this too hard, but this is just a timeline of when things develop after an individual is conceived. So you can see that early on things are starting to happen even 6 to 7 weeks. EGA stands for, I believe it's estimated gestational age. Estimated gestational age. So it's basically once the sperm and the ovum unite and conception happens, from then on, going by week, things happen as that new individual is forming. And so friction ridges start forming relatively early. Volar pads are excuse me, I got something in my throat. The fatty parts of the palms and fingers that develop first, that then lay the groundwork for ridges to develop later on. And ridges start to form as early as, I believe it's 10.5 weeks, if my memory serves me. And they can go all the way up. So primary ridges start forming all the way up until 16 weeks of EGA, or estimated gestational age. And sweat glands start forming, and you can take a look at this, and I can also show this as well. So it's just basically what I'm trying to say is that as we're going in, friction ridge starts developing relatively early in an individual's development, and then stops. So secondary ridge formation and furrows start finishing and maturing all the way up into 16 to 24 weeks of estimated gestational age. So it's a relatively quick process that your fingerprints are formed. And from that point on, like we talked about before, they're going to stay persistent all the way until death, unless something happens as far as damage or disease or the like. There are two important timing events that happen that also affect pattern types and ridge formation and those kind of things. I won't hit it too hard, but this is also in that paper that I had mentioned before, the Wertheim and Maceo article from 2002. There's two timing events. The first one is when sorry, I lost my train of thought there for a second. Oh, that it's the onset of those volar pads. That's it's kind of like a bulbous fatty area that starts to develop when that starts to regress. And then when the other timing event is when friction ridges start to form. And so when those volar pads start to regress and when the friction ridges start to form, determines how the pattern type is going to be. Okay, so like ridge counts, what ridge counts are is, so if you look at the center of your finger, there's a core, And a lot of times you're going to have a delta where it's an area where 3 ridge flows come together and create a delta or a triangular looking formation. And so a ridge count is from that core to that delta. And depending on when these 2 timing events happen, that ridge count could either be large or small. And that can also affect ridge flow and when minutia are produced, as if you have a larger core area, you may or may not have more minutia versus a smaller core area, right? Just quickly, I'll mention this too, because this is kind of more for people that maybe do a little bit more of the comparison or have a little bit more experience with fingerprints, is there's a little bit of debate on whether or not ridges form in pieces called friction ridge units, or if ridges themselves form like as a unit and form as a ridge before just an individual ridge unit. And I mentioned the source book here. That's that one is a free resource that you can use. It's called the Fingerprint source book. The U.S., I want to say it was the U.S. Department of Justice created that book. It's a PDF that was created, I think, in 2012 or 2011. But I can try to see if I can't send that link to the PDF, or I might have it at the end of this slide, or sorry, at the end of the presentation. But that's a good one to read through too, and they just talk a little bit about that controversy, if you will, between whether or not ridges form in units or they form as a, meaning that it's one ridge unit versus a full-on ridge as it's developing. And the minutia formation models, we kind of briefly talked about a little bit, the Turing model or even the Merkel cell model, where again, Merkel cells start to line up along stressors because again, Merkel cells are pressure, tactile. And so as your finger is developing, tactileness or pressure along your finger then lays down in a specific way that then can lead to minutia formation. So ridge formations start in 3 areas. So the apex or the core, the middle of the finger, the distal or the tip area, or down towards where the distal portion of the finger meets the middle portion of the finger. And so depending on those 2 timing events we were talking about for the volar pad regression and the ridge formation can determine whether or not the ridges start forming more in the middle or more towards the top. And so that can create, as you can see as you'll see here in a couple slides, I'll show that in a minute, how big or small that core might be, what pattern type it might be, depends on where that volar pad is located. And so then they all will meet in the middle. And like I said, so this is a core or the center of it. And then these right here are called your deltas, where you can see how you have 3 ridge formations. So you have this ridge formation up here on the top, the one on the bottom, and the one towards the middle. They meet, and where they meet creates a triangular looking shape, which is your delta. And so you have 2 of those on either side of this. This is a whorl pattern type. And so here you can see a little bit more what I was talking about with the timing events. So depending on how big, so the volar pad is like a swelling of the skin that creates like a bulbous or a fatty type of structure. And if that is still fully developed when the ridges start forming, you're going to have a big or a more even flow whorl, okay, with deltas that are way off to the side, as you can see in this first one, the image on the left. And then as that swelling is smaller, once the ridge formation finishes, determines how close or how big that whorl pattern gets. And you can kind of see as it's diminishing, you go from a really big swelling in that volar pad to nothing. And then that's where you're going to see a more arch pattern because there's no circular formation from that swelling of the volar pad. Versus even one right here, the second from the end on the right has a very small core whorl pattern because that swelling was very small. And you can see what a fingerprint would look like with that. Okay. And then symmetry, as far as that volar pad, makes sense as well, as far as whether or not it could be more of a loop versus a whorl versus an arch. Typically, arches are because those volar pads, which again, like I said, they form that bulbous swelling on the fingertips and sometimes in the palm. If those have completely regressed or no longer around, once the ridges start to form, that's where you're going to get a very simple, easy flow arch pattern. Okay. With the symmetry, like I was talking about, if the symmetry of this volar pad tends to lean let's say it leans to the right or to the left, then you're going to get a loop formation, or tend to get more of a loop formation. And again, the size also depends on how big that swelling is. So this one, swelling is big, but it's leaning towards the right, and that creates that slant loop. In this case, it's a right slant loop. And so you can see again from here, as the swelling or the volar pad, sorry, I lost my train of thought. The swelling, the volar pad starts to regress, but it's still leaning towards one side. Then you're going to have smaller and smaller loops to, again, where you get one that doesn't have any of the volar pad present, that creates an arch pattern. Okay. Asymmetry is a continuum, so it can that asymmetry might be subtle, and so you might have a leaning whirl of some sort, but then it can be as much as all the way to the side where you're getting like a really big loop. So that's it can be very subtle changes, and also the volar pads themselves, as far as the size of them, can be very very gradual and across the continuum as well. So that's why you're going to see a whole host of sizes and leanings and all that kind of stuff between all the whirls, loops, and arches that you're going to see, and that creates the really cool variable fingerprint patterns that you're going to see across the human population. If you haven't had a chance to look at a lot of fingerprint pattern types, there's quite a few out there. It doesn't just stick into one very specific plain pattern. You're going to see, like I said, big loops that have, like you can see on the image on the far left, where the delta is way off to the side, or you might see something really small like this one on the right side here. This very first looping one next to the arch that has a very small loop pattern to it. All right, quickly we'll go over damage and wound, and that should probably bring us to the end. We can go over some of those questions. So as we mentioned, the friction-ridged skin persists throughout a person's life. Injuries can disrupt that as long as it reaches that basal layer. Wound healing happens in 3 phases. The first phase is an inflammation phase, then it's followed by the proliferation and tissue formation, and then there's the last one, which is the tissue rebuilding. And this image right here is an image of a scar. So scars are usually visible because ridges, as opposed to continue going, they kind of turn in on themselves, and kind of see like what's called like a puckering, where you see the ridges kind of turn in on itself. You see this line right here that is created when the ridges kind of pull in on itself, and you'll see why that happens, because once the cut happens, the body tries to pull the 2 ends together and then cover that area, and I'll show you what I mean by that in a second. So the first phase is the inflammation phase. So it's the immediate response, and so you can see as we're going down through here for the different figures. So at first figure, and these images are all from the Fingerprint source book in chapter 2. So again, I should have I think I have at least the QR code to reach that, and then I can try to send you the link as well if you can't get it from the QR code. But that top image is intact, normal skin, and then some kind of injury happens that reaches down into the dermis. So you can see how it reaches down into the dermis right here. The dermis is this pink area right here. If it had just been a cut that only goes right here, it would just be a few weeks later, once the epidermis regenerates, everything would be good. But this one was cut way down into the dermis, and so immediately you're going to have an immediate response. Blood is going to start pouring into that wound. I mean, if you've ever cut yourself, you know what happens as soon as you cut yourself. Shortly thereafter, you start seeing blood show up. Okay, the reason for that is there's platelets in your blood that will help with the clotting, and they also the blood also helps recruit immune cells to help fight off any bacteria or other things that might be trying to invade your body at that point, because now it has access to your blood supply and can start getting deep into your body. Immune cells will immediately start killing off any bacteria and remove some of those dead cells. And then the dermal cells will concentrate on wound healing or helping with that, and repair starts going underneath that blood clot. And for purposes of these images, just assume that there's blood and blood clot over the wound. The images are just kind of showing what's going on underneath that blood clot, but for the illustration purposes, we're showing it without the blood clot, but it would normally be there, like it would start scabbing over or having some kind of a cover to protect yourself as it's healing underneath. So moving on to the next phase. So that first phase is just going on right there. It's that initial cut, blood shows up, it starts trying to heal itself. The second phase is the biggest one. A lot's going on in this particular phase. So the basal keratinocytes are those basal cells, those basal layer cells, the base layer cells are the ones that are going to start taking control, and they're going to start going through some changes to then work on covering that area to get yourself back into normal form. So what will start to happen is the desmosomes and hemidesmosomes will start to dissolve, and certain filaments and pseudopodia will start to extend. So the pseudopodia is it's cells that will start moving across from both ends to try to work to get those two ends to connect to each other. And basal cells will start to divide and cover that area. So you can see from that top image, the one that says figure 225, the basal cells are starting to contract. Your skin is starting to contract in. The basal cells are moving and contracting towards each other, and eventually they'll meet in the middle to cover everything, to then start working on building structures above where they've meet. And like you see, like the very last bullet point, the dermis contracts the wound to shorten the distance. And because it contracts to shorten that distance, that's where you're getting that puckering that I was talking about before in this image. You can see right here where this cut was down the side of this finger, and this side came together to then meet in the middle to cover the wound faster by doing that. And that's what I was talking about before. So that contraction makes that classic puckering. That doesn't happen every time that you're going to see that puckering from a scar, but that's typically what you'll see with a cut that went deep enough. So yeah, so those leading cells in that cell migration will reach each other, and then they'll once they reach each other, they're going to start reestablishing communication and connecting to each other by those gap junctions, because they need to be able to communicate from one side to the other so they can coordinate what they're going to do as far as rebuilding itself and moving forward. And then once that basement membrane has been established, then they'll start sending cells up just like before. That they'll start filling in the gap in the epidermis as it's moving forward. Once there's enough of an appropriate layer that's been created, that scab will fall off and everything will kind of get back to normal. The basal layer will now have its new template, or its new surface template, because remember before that I said that as long as the basal layer stays the same, it'll keep producing cells upwards that will keep the same skin structure as before. Well, now since it's been disrupted, a new template has been created, and now everything going forward will look like that new template. And so that's why scars, that when they disrupt the ridge flow and create that puckering, now going forward, that structure is going to be the one that is going to continue to reproduce every time the skin cells create new ones as it's moving towards the epidermis and going towards the surface. Because those primary and secondary ridges were removed, you're not going to have the ridges and furrows like you would normally see. And so like I said, that's when the scarring starts showing up, and it creates a whole new structure, a whole new ridge flow, because there's not that underlying structure that we were seeing before. And if there were any sweat glands or connection to sweat glands that were cut or removed during that wound, they will also be covered over, and you won't have any sweat glands in that area. Potentially even any pores in that area as well. So it's creating a whole new structure that's happening now. That structure is going to stay persistent throughout that person's life, unless again another wound happens that then would restructure everything again. And then the last phase, the shortest phase, after the epidermis has reformed, it's going to start focusing on the dermis. So the epidermis is the priority, because we want to make sure that the outer layer is covered and protected, so we're not allowing or introducing any more bacteria than necessary. And then they'll start focusing on the dermis, and then the dermis will go through its remodel and reinforce any kind of scar tissue or things that are happening underneath that injury to just bring everything back to normal. And then here's just some other views of similar thing that's going on. So it has a kind of a side view as well as a view from top down of what it looks like. So the very top one on the left, nothing happens. Then the cut, you can see it reaches down into that dermal layer. So that's what that pink represents. It reaches all the way down into here, and you can see how deep that wound was cut. And then you start to see it starting to create and moving to link up with itself to then cover that wound, and it starts to pinch things in to cover everything as it's going through and just rebuilding all those different layers again once the connection has been reestablished and it starts to cover itself. And again, this is also showing without the scab present, just to give you an idea of what it would look like if you can see through that scab that was formed. Here's the QR code for the fingerprint source book. If you can't get this or need me to send the link, I'll try to work with Krino Forensics to get you guys the link, as well as for the references that I have here on the next slide. I'll give another moment here for you all to look at that QR code and get it scanned as we start moving forward and then get into the Q&A section. So if you have any other questions, that's what I have for you today as far as everything. So if you have any other additional questions, put them in the chat and we'll try to see if I can't get them answered as best I can. I'll wait just one more moment here for the source book, and then let me start looking at some of these questions as I wait for y'all to get there. And I'll show you the references. Here's the references that I use. I've got a couple pages of this. And again, I'll try to, any of these questions or any of these references you want or need or just need some guidance on, I'd be happy to help with that. And then here's the second page of those. Some of these might be open source, so you wouldn't have a hard time getting them. But I think a lot of them are not open source, so I apologize on that front here. Let's go back to that first page. I'm trying to remember if any of these are open source or not. But if they're not open source, hopefully you have some access through a university library or some other avenue with which to reach them. All right, well, I'm going to stop sharing so I can start looking at some of these questions that we can start going with. So think about those questions that you have. And if something comes up that I want to show the slideshow again, maybe I'll pop that up. Let me get my questions. Sorry, I got to get back to the... And also too, I mean, if it's easier, you potentially could go off mute and ask that way, where are my questions at? Sorry, I don't know Google Meet as much. Where do I get to the chat? Right here? Oh, here we go. Sorry, I don't tend to use Google Meet. I use Microsoft Teams a little bit more than this. Let me just scroll through these questions here really quick and see if I can answer them. Okay, so the very first one, and if I mispronounce your name, I completely apologize. Shrivali, the question is, there's a recent case in India. I've heard about this case, but I haven't been able to look at it very closely. So if anyone has more information on it than I do, I'm all ears. I'd be super curious to hear about it. About 2 children that were reported to have similar friction ridge patterns. Until I can look at them, I don't know if that's true or not. I would probably lean on the side that their patterns are probably very similar, because like we were talking about before, when we're talking twins, genetically and environmentally, they developed in a very similar fashion. And so in a lot of twin studies, twins do tend to have very similar pattern types. But when you look at the fine detail, the minutia, and get into the the more microscopic level, you can distinguish one from the other. Now, the biggest question comes if one or both of those twins were to leave a fingerprint at a crime scene, and if that fingerprint is not a very good quality fingerprint, it might be, due to the quality of that fingerprint, very difficult to distinguish the one from the other twin, right? So that's where I say that uniqueness is kind of a moot point, because while their overall fingerprints are different, when we're talking about crime scene prints or prints that were found at a crime scene, due to how good or not good that fingerprint is, we might not be able to distinguish the twins from each other, right? Let's see whether there's more. How do forensic experts address such cases, and what additional methods are used to ensure accurate identification? I mean, I would hope, so getting into how we address such a case if one were to exist. Now, like I said before, we'll never be able to fully confirm that uniqueness is true across all of human history. And even if we did find 2 individuals that had the exact same fingerprint, let's just throw that out there and say, let's just say that there's a possibility, or say that it is true in this scenario. I don't think it negates the importance of using fingerprints for comparison purposes, because if it's just one set of individuals, every other person or every other case could still be used. Now, in this situation, if we came across something like that, I would hope that whoever is investigating, like say the police officer or whoever is doing the investigation, once I've completed my comparison, could hopefully explain that this came from twins. You know, do we need more information from them? And that's where using things like a likelihood ratio could be helpful, because I'm not saying that this particular fingerprint is from that person. I'm saying that the likelihood of it being from this person is greater than it from being from somebody else, based on the information that I have, or the evidence that I have is more likely to be from the one person than from the other. The likelihood ratio would then be a lot lower, assuming twins. And so with a lower likelihood ratio, that might then make me back off and think that I need to be a little bit more careful with how I present this if I were had to testify. I don't know if that's really answering your question or not, but there are other things that we use as well where we might do. So the current methodology that we use across fingerprint comparison, I think this is pretty uniform across multiple countries, is what's called the ACE-V methodology, and it stands for analysis, comparison, evaluation, and verification. And so if I'm the original examiner, I'll go through and I'll analyze the fingerprint, mark whatever minutia I'm seeing, pattern types, ridge flows, other things like creases or other features within a fingerprint. Then I will compare that fingerprint once I've completed that analysis to, two individuals in the case. And then at that point, I will evaluate again the evidence and whether or not the evidence shows more support for it being from the person I'm comparing it to, or more support for being from somebody else. Okay. And then after I'm done with my examination, it'll go to an additional person to do their own analysis, comparison, and evaluation. And in certain situations, we could do what's called a blind verification, where we just send the images to somebody else without telling them anything about the case. And so that's just a way to have a quality measure in place in hopes to mitigate any errors that might happen. But with this situation, it's tricky, because I don't know how we would be able to go about this if in fact two individuals shared very similar fingerprints. I hope that answered your question enough. If it didn't, add another additional question at the back end or at the bottom of this, and we'll see what we got. We talked about twins and fingerprints, so I won't go much into that unless you have more specific questions in regards to that. Okay, we already talked about identical twins and the ones in India. And yes, like we talked about, identical twins will have different fingerprints. Again, looking at the whole finger, the palm, everything, they will be different in some capacity. Okay, here is a question in regards to the automated fingerprint identification system, or known as AFIS. It asks, with the advancement of technologies such as AFIS and large biometric databases storing fingerprint data, how real is the risk of biometric data breaches? In case such data is compromised, can fingerprint information be misused or replicated? That is a very good question that I don't know if I've ever come across in any of the research that I have done or the articles that I've talked about, because we don't tend to, as a fingerprint examiner, it's not really my place or worry per se as far as data breaches to some of these databases. But that's actually a really interesting question that I don't know if I can fully answer that for you. That's interesting. I don't know of any cases of individuals breaching those, because a lot of times, for example, in the agency that I used to work for in the state of Idaho within the United States, is in order to access these databases, you have to have a special link that doesn't go through a normal internet. And so it makes it much more difficult. It has a direct pipeline to these databases, so it would be very difficult to breach them. So that's probably the only thing I would probably be able to add with that, is that due to the engineering structures that they have in place, meaning that you have to have specific terminals or specific computers that directly link to those databases, That's really the only way you can access them. Same thing with like the FBI, that there are specific computers and terminals that you have to use to access that. But since I don't work for any of those companies, I don't know what they do on their end to protect those databases. So that's actually a good question. I apologize, I don't have a better answer for you on that one. As far as misuse and replicated, I mean, that can happen in the sense that if someone were to take a mold of your finger and put, create a mold and then create a stamp, you could then potentially stamp someone's finger on evidence, and then that could cause investigators to start looking at one person that had absolutely nothing to do with the case as opposed to one that actually did. That's one of the only times I can think of at this point of using replicated fingerprints, or even taking someone's fingerprint from images they find on social media from Facebook or Instagram or stuff like that. So that's why you shouldn't take pictures of your hand, because someone could potentially take a digital image of it and use it for bad purposes and like. Oh, good question. I'm sorry I missed this. I missed this during my presentations. Question 5. It's asking if a person has a temporary cut on their fingerprint during a crime, but it heals later. How do examiners handle such changes during comparison? That's a good question. We like to try to get the most current known fingerprint from that person, but that doesn't always happen. We can't always tell when we're looking at a crime scene print whether or not there's a cut present, because a lot of times those cut just show up as like gaps or lines in the fingerprint, and that could be due to a crease, or it could be from a cut. I will try to mark those when I'm doing my analysis, because I'll use things like a Photoshop to where I can digitally mark something and use different colors to indicate where maybe like a crease is or a potential scar or cut, and then just make sure I note that going forward. But hopefully there's enough other information beyond that cut that I can then use to compare to that person. But then if I do, or I am able to get a known fingerprint card from that person around the same time as the crime, that's just another piece of information that I can use in that likelihood ratio to then link that fingerprint to that particular individual in the case. I hope that answers what you're looking for. We try to take every piece of information we can at the time. Sometimes we just don't know if it's a cut until we've actually looked at the card, and then at that point we can make a guess or a thought on that. When we're doing our analysis, we'll look at the latent print or the fingerprint from the crime scene first, so that we're not biased from looking at the known impression first to be like, okay, oh, there is a cut right there, that's this line right here, when in fact maybe it is from somebody else. And so by having that biased information, I might be more prone to error. Okay. I hope that answers your question. Let me know if you have more questions on that. We already answered about the sweat chemistry. If I didn't answer that one fully, let me know. What are the best methods to collect and preserve latent and patent fingerprints, and what alternatives exist to lifting tape? This is a tough question to answer, because there's a lot of ways to collect and preserve fingerprints. The main one that I would use is probably just digital photography. I think a lot of times you can photograph a latent print pretty well on almost any surface, and so I would definitely start with digital photography. If it's on, say, like a piece of paper, that fingerprint, I could scan it. If I had a really good quality scanner, I could throw that paper or whatever it is on a scanner and then scan it that way at a high resolution. Then of course there's the powder and lift technique, where you can powder an item and then lift it using tape. But then there's also certain things that are almost like a putty type stuff. There's this technique called microsil that is a putty type stuff that you can mix together. It has I forget what it's called, but it has two components to it. You mix them together and it creates a putty type material that eventually hardens, but it doesn't harden so hard that it still has a little bit of some give to it, so that you can go on like a curved surface or a more textured surface. It fills the gaps a little bit better and then can pull that fingerprint in powder off of that surface. And then there's another technique or product called AccuTrans. That's a very similar type thing. It comes in this gun that's about this long, and you squeeze it together in this gun that then puts the two chemicals together, that then again creates like a hardening material putty type stuff that then covers the surface and lifts that way. But those are probably I would probably start with photography. I think you can do a lot with photography. You just have to be patient take your time, use a manual camera to where you're adjusting for the shutter speed, you're adjusting for the aperture, you're accounting for various different things. It's much more difficult, and it's a skill that requires some time and practice, but you can get really good at photography. And a lot of times you can preserve prints very easily with photography. Okay, let me know if you have more questions on that one, if I missed it. Okay, the next question, in real crime scene investigations, what would you say is the biggest challenge you face while developing, preserving, and collecting latent fingerprints, especially under non-ideal or contaminated conditions? That's a good question. I think while at the crime scene, because I tended to be more in the lab, but for a number of years I did go to the actual crime scene itself, and I would say some of the biggest challenges at the actual scene is just being in the environment and having to potentially be there for several hours at a time. I went to one scene where I left my house at 9 p.m. and got to the crime scene at about 10 or 10:30 p.m. And then overnight, all the way into the next morning, I was at the scene. And so I was essentially awake because I went to work earlier in the day before I left work. And then they called me to go to a scene in that evening. And then I spent the whole night into the next morning, all the way to like 6 or 7 a.m. the next day at that scene. And so it's just the amount of time sometimes the scenes take and the environment, and whether or not the weather has to play into whether it's too hot or too cold or rainy or dusty or various other things. And then you also have to worry about, when you're at scenes, people that are out and about wondering what you're doing. You hope that the police that are there assisting you can help cordon off an area, that you don't have people entering the scene that shouldn't be. But that's also another one too, is people entering the scene that you don't want in there. As far as in the lab, some of the biggest challenges is just figuring out what's the best way to develop fingerprints that you can't see. Because remember, latent prints or crime scene prints a lot of times need some kind of chemical or light or physical technique to be able to see it. And sometimes it's just figuring out what's the best technique to use for that particular item, and then what's the best technique for collecting, like we were just talking about, and preserving that, whether it's photography or lifting it or things like that. And curved surfaces sometimes are the hardest one to photograph. And so you might be photographing the same latent print 15, 20 times or more. And that can just be really frustrating because you're trying to get the angle of the light just right. If you have a flashlight in your hand, you're angling the light to try to get the fingerprint to show up. And so that can be really a daunting task, and trying to figure out, how do I collect that and preserve it the best way. So I would say those are probably some of the bigger challenges. I'm sure there are more, but those are the ones that just came to my mind quickly. So the next question, is there any case a person with immigration delay syndrome committing a crime, and how their data is recorded in the crime scene records? Very good question. I don't know of any situation. So let me backtrack. So what the immigration delay disease is that there are certain people, I don't remember what the number of it is, but there are certain people that, due to their biology, due to their genetics, they did not develop that friction-ridged skin on their hands and feet. And so they essentially have smooth skin, much like you would find on your arm or your leg or whatever. So they don't have friction-ridged skin in the way that we know it. Now, as far as them leaving fingerprints behind, I don't know what that looks like, as far as would their skin show some kind of anomalous or features that could be variable to them as an individual, more so than like the ridges that most people would tend to see. But as far as crimes that someone with this disease has, I honestly don't know if there's been a recorded one of that. And I have definitely I've heard of situations that there is an article, I can't remember the title of it, where someone who works a lot with their hands and a lot of chemicals, the chemicals changed the way that their fingerprints looked, and that person committed a crime, and it did not look like a normal fingerprint. It wouldn't be like the immigration delay disease per se, but it showed very patchy, I don't know how to describe it, look to it. But again, I would make sure to note a lot of things as much as I possibly could during my analysis, to show what I might have. And at that point, if there isn't, or if there aren't features that I feel like I can use that are variable enough or have enough selectivity, I might just say that that particular fingerprint is not suitable for comparison, and then I might not ever compare that to anyone in the case. I'm trying to think of other ways to describe that, but I think that's the best way I can think of it this time. I don't know of any situations of someone doing that. I do know there have been a couple of cases of people trying to, say, for example, enter the United States, but couldn't because they didn't have fingerprints, and so they couldn't do a background check on that individual to see if they've committed crimes in the past, which is why it's called immigration delay disease, because immigration offices use fingerprints to determine if someone is who they say they are and if they've done anything bad in the past. Moving on to the next one, how does environment, exposure, heat, water, time affect fingerprint quality? Good question. I mean, it can affect a lot of things on whether or not the strength of the ridges show up, whether or not they're faded, if it rinses away some of the chemicals that we use, or I should say for example, we use amino acids for to develop fingerprints using things like anhydrine or indanedione, and if water or environment removes some of those or makes it much more difficult to develop those, that can definitely have an impact on quality. So everything environment-wise can play on it. So whether or not it rubs off the ridge flow, the residue that's on the surface, heat can definitely start to degrade the ridges. But also, too, on the flip side of that, depending on how the fingerprint is deposited, there have been cases where fingerprints have lasted for years on the exterior of a surface, like an exterior window or a door that is exposed to the outside, just because of, if it's like that more sebaceous material behind like your ear or on your forehead, sometimes those fingerprints can last quite a long time and don't have as big of an impact from heat, water, and the environment. There's one case out of, I think it's out of the UK, out of the United Kingdom, where someone was identified in a case off of the automated fingerprint identification system that was from a case, and the individual that was found was like, I've never been to this location. The only thing I can think of is 10 years ago, I assisted in building like a house or a window or something like that in the area. It's possible it could be from that. And so there seems to be a situation there that he was using some kind of maybe residue or sebaceous material that just lasted a long time. And because we had this idea, or at least in the past, we had this idea that fresh prints tend to be the strongest one that we develop with, say, like powder, that doesn't have to be the case. That really strong prints could be from oily residues or from some kind of material that you use during building processes and things like that. So environmental effects can have huge impacts, or they might not have any, depending on what the fingerprint is made of. Let me know if you have more questions about that. This next question, how do injuries like cuts, scars, or abrasions affect fingerprint patterns? We kind of talked about that a little bit. They can definitely disrupt the ridge flow and pattern type. If you get a cut across that core of your finger, if it's deep enough, and because like we talked about, when the ridges when the skin tries to start fixing itself and it comes together to reattach itself, that can cause a very prominent whorl pattern to look totally different, or even a loop pattern to look like a whorl pattern. So it can have a big impact on the way pattern types and ridge flows work with those scars and injuries and things like that. So next question, for a week-old latent fingerprint on a wet glass bottle, which development method would you choose and why? That's a good question. There is a technique called SPR, and I'm drawing a blank as far as what the SPR stands for, but it's essentially it's in the category of what we call powder suspension. So it's a fingerprint powder that's inside of a liquid and has a surfactant that allows it to stay in solution, and you can spray it across a surface with like a spray bottle, and it can be wet or wet at the moment, and you can still develop it on that surface. So I would probably say an SPR type of a thing. Or if it's dried, I still might use my normal sequence, which is super glue followed by a dye stain followed by powder. But SPR is a really good technique for wet surfaces that are non-porous, like this glass bottle would be. So yeah, it's if I knew it was wet, or it was wet at the moment, or I was at a scene and the car was wet and I was doing the exterior, I would use SPR. And I apologize, I can't remember what SPR stands for. But it's a powder solution like that. Next question, what are the most significant recent advancements in fingerprint technology? That's a good question that I think we're still working on a lot of those advancements, and we tend to be a little bit slower on picking up some of those advancements. I talked a little bit about likelihood ratios. That's a very good example of using like a statistical model to develop those likelihood ratios. Those are in the works now, and some have been created, and I would argue them as being a good advancement, but people within my community don't like to use those because they don't feel like they provide as much information as an individual who's been trained fully to do the comparison. So I would lean more towards that, but we're also starting to get into a lot more technology that's using visual aspects, like taking advantage of the light spectrum, using the ultraviolet spectrum and the infrared spectrum to develop prints more, because there's the visible spectrum, which we can all see with our eyes, but using some of these ultraviolet or infrared spectrums, we can still develop fingerprints that we normally wouldn't be able to see using these technologies. So that's another one I think is developing quite well at the moment. So I'm sorry I don't have a very good most significant. I would probably lean more towards the quantitative methods as a technology that's being developed, but also these quote unquote non-invasive visual techniques that are being developed are interesting. And I haven't had enough time to review them myself, but I think that's the way a lot of people are wanting to go, because you're not applying chemicals that could maybe not degrade the fingerprint, but you can use these techniques with these light sources, photograph it with the light source, and then move on to your chemicals. And so you can maybe get images quicker or faster, or preserve something before adding chemicals that always has the chance of affecting the latent print if you do it incorrectly. Sorry that the video didn't work before. I'll try to see if I can get you the link to that sweat pour video. So it says that the identical twin state, or the identical twins that from before that we're talking about in India there, was stated as identical. I would really like to see those images. So if anyone has or knows anybody that can get a hand on those images, the fingerprint community would love to see that. I know of people that would love to see that. So if anyone has any inside information or know of how to do that, that would be amazing. Or if you could write up an article on the images, that would be awesome if you could do that. You get a lot of, I think you get a lot of feedback from the latent print community if someone could write up an article showing the two images, or showing, let's just say, even if it's just this finger, you know, one of the index fingers, or all 10 of the fingers, if you can get a hold of that, that would be really interesting. Or some kind of a presentation, that would be amazing. I would love to see that. I wish I had more. And it looks like the last question, so if you got some more, get them in the chat. But the last question here, which amino acid is more abundant in the fingerprint residue? Ooh, I'd have to go back and look on that one. I don't recall off the top of my head which one is the most abundant. There are a few of them that are in the sweat, that when the sweat reaches the surface and it touches your fingers, that's what you're leaving behind a lot of times in especially when you touch glass or paper, more so paper. And ninhydrin and indane and another chemical called DFO, which is diazofluorineinone. Those react with the amino acids. As far as which ones, I'm sorry, I can't remember off the top of my head which exact amino acids is the most abundant. I'm sorry on that one. It very well could be serine. I just don't remember. Let me see if give me a minute. Let me see if I can find one of my articles. I think I know where to find it. Give me 1 second. I'll give you a minute here to. Okay, so someone else. So thank you. Is it Natish? Sorry if I mispronounced that, but according to, he's saying it's serine followed by glycine. That sounds right. I don't always commit that to memory because that's not, you know, I don't get that question very often, if ever, because we just want to know, you know, if it is amino acids, I want to use something like anhydrine or inanedione to then enhance or develop that fingerprint, right? All right. Well, I'll wait a few more minutes here if anyone has any other questions, but I'm all ears for questions. Let me see if I can get the link to the fingerprint source book. I can put it in the chat. That might help right now. Let me see if I can find it. If not, you can always just search fingerprint source book. But I've got the link right here. I'll put that in the chat right now. So that's for the fingerprint source book, and then here's it's a YouTube video that I was showing before. Whoops, no, sorry, I just made a mistake. Hold on here. My apologies. And then here's the link to the YouTube video that I showed before about the sweat pores for those of you that weren't able to see it. And then just let me know if someone else needs those links going forward, or if you want any of the links to the art, well, not links, but if you want the references I said before, I was showing before. Like I said, many of them are probably not open source, so if you don't have access to them, I apologize. One area, I got one more website I'll put up here that you guys can check out too. There's a library, a research forensic library that's housed at a university in Florida that you might be able to get access to some of these articles because they were able to get them and provide them for people as well. So you can try out this one. That's the forensic library. It's at the Florida International University out of Miami, and they might have some of those articles in there that are free to use. Now, I haven't looked through this to see which ones of the references I used are in there, but you can at least check that out. Or there might be other references in there that you might be interested in going forward for any kind of things or anything like that. Oh, sorry, that's probably my YouTube. All right, sorry, that's my YouTube started going on to the next video. I apologize for that. So if you don't have any other questions, like I said, I'll hang around for a minute. And okay, yeah, one last question. How the report is written after a fingerprint examination, like the points match, will we write 100% match? I won't know. So what the report would say, and they've changed over the course of the years here, but right now, the reports that I would write would say something like latent print or fingerprint number whatever or letter whatever. So we have to make sure that we mark what the fingerprint is. So when we're comparing it and putting it into a report, we can identify which fingerprint we're referring to. So let's just say fingerprint 1.1 is identified to the, let's just say, the right thumb from John Doe or whoever the individual is. And then in the body of the report underneath where I said like this latent print was identified to this finger to this person, I might then make sure to clarify what I mean by identification. So identification right now, for me, and then there's a standards body that I should show you that as well. There's a standard that talks about how when we're talking about identification, what it means is that the data or the comparison information, the evidence supports or there's more support for this fingerprint being from this individual than from somebody else, and that I would not expect to find this amount of correspondence between two people that are from, no, sorry, let me rephrase that. I wouldn't expect to find this amount of correspondence between a unknown fingerprint and another individual, okay? So I'm saying that the data or the comparison I just completed shows more support for it being from the individual I've listed than from somebody else. And I wouldn't expect somebody else to have a similar arrangement of features. Okay, that's how I would say. I would never say it's 100% match. We cannot say 100% match, especially when we're talking about science. We can't say with complete certainty that it's this person, because as we were talking about before, depending on the quality of the fingerprint, there could be someone that has very similar features that, because of the quality, I can't distinguish one person from the other. I'm not saying that two people have the same fingerprints. I'm just saying just because of the quality of the fingerprint, there could always be someone that confuses me or others to it being that person when it was in fact from somebody else. Okay. Okay. In the United States, we wouldn't write it in that sense that it's similar or exact match. We wouldn't write it as match. We would just say it's been identified, and then, like I said, we use clarifying language to say what we mean by identification. And also, too, is there's a recent standard, and I'll pop that up here too for you to see. If you go to, let's see, hold on, bear with me 1 second. I gotta remember where I'm going. There's a standard that was developed by a board of individuals. I'm on one of the boards that talks about what are the different conclusions, whether identification, exclusion, and stuff like that, that we can use when we're determining or deciding on fingerprints. And sorry, bear with me one more quick second. Here's another link to check out. This is the current standard for the conclusions that we use within the United States for conclusions about a latent print to individuals in a case. So we could say there's a source identification, but we can also say that the comparison is inconclusive, but there are some similarities with the person I'm comparing to. So what that basically means is I don't have enough support to reach a source identification, but there's enough to say that there's something going on here that, I think this person has the potential to be the person that left the latent print, I guess is what I would probably say. That we're talking about support for it being from somebody else, or sorry, support for being from one person over the other. So we're using the evidence, the data, the number of minutia and things like that in correspondence. That's going to support more it being from the person I'm comparing to than from somebody else. And so depending on how much support you have, that goes down from source identification to the next category, which would be inconclusive with similarities, to a straight inconclusive, where when I look at this latent print or this fingerprint, I can't determine whether I think it is or isn't from that person. And then it goes from there whether or not I think I can exclude that person or not. And so that last link, some of it might be difficult to understand for those of you that are newer to this particular field, but that's the current standard that we use for the conclusions that we use in comparison. And to gain access to that standard, you have to give your email address. But as soon as you give your email address, they'll give you the link to download that particular standard. And those standards get updated every 5 years. And so that one was just published last year, and so it will come back around the cycle in a couple of years here to make any updates to it. And maybe we've changed our conclusions by then or not. But currently, right now, we're using more of a likelihood ratio. It's a descriptive likelihood ratio as opposed to a quantitative one. We're using words as opposed to numbers at this point, but it still is a likelihood ratio. We will never say that this fingerprint originated from this person. We'll just say that, like I said before, the evidence supports or more supports it being from the person of interest than from some other person. Okay, I hope that makes sense. Some of it's hard to understand, and I apologize for those of you that are newer if you have questions or confusion on that. But like I said, I'll stick around just for a few more minutes if there's anything else. Otherwise, I really appreciate the opportunity to come present to you guys, to your group. I hope you learned something from it. I hope I didn't go too fast or was too confusing. Feel free to reach out. I will do one more quick thing here. I'll just share some information with you. I'm going to share the screen really quick again. Bear with me one quick second. Where is my... So this is just like I just created this flyer to put up for. Oh, hold on, I got to put it into presentation mode. Hopefully, well, you can at least see it right here, anyways. Can you see? This is just a little flyer I put together for my workshops when I go to the conference. This just has my email address if you want to reach out to me, and some of the things I can do for agencies if they need help with training or processing latent prints or doing any kind of comparison type stuff. So feel free to reach out to me if you have questions in the future or have questions about articles or research or things like that. I'm more than happy to help. Y'all can see that, right? Can everyone see it? You give me a thumbs up if you can see it all right. Sometimes, like I said, I'm new to Google Meet, and so I'm not quite sure. Okay, thanks. I'm not quite sure when things show up effectively or not. So yeah, feel free to ask me questions in the future if it comes up. And but other than that, I don't know if anyone else has anything they want to bring up, or if y'all have any other announcements you need to make for the group. I'll turn it back over.
Thank you, Mr. Craven, for patiently addressing the questions and sharing your valuable insights with us. At this moment, on behalf of Krino Forensics, we would like to formally present you a letter of acknowledgement. Sir, as a token of our gratitude for your contribution to this international academic forum, the acknowledgement letter will be shared to you digitally after the session. Okay, sir? As we come to the conclusion of today's webinar,
Thank you.
I extend our heartfelt thanks to Mr. Nicolas Craven for generously sharing his expertise, experience, and time with us. We are grateful to all the participants for your active presence, thoughtful questions, and enthusiasm throughout the sessions. E-certificate will be shared with all eligible participants after verification. We encourage you to stay connected with Krino Forensics for more such academic and professional engagements in future. Thank you once again for joining us today. We wish you a wonderful day ahead. Thank you, sir.
Thank you all. Have a good rest of your day.
Thank you,
Thank you so
sir.
much, sir.